There is power in the name of Jesus. Yes, there is power, oh, in the name of Jesus. To break every chain. God, you're a chain breaker, yes you are. To break every chain.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started. All right, looks like we are live. Yes, it's late night on SFT, but we did have a good lecture earlier with Brother Sal, which was earlier than I guess we typically stream. Good people in the audience, in his image. Good to see ya. Great video today debunking biologos. Make sure, guys, you go subscribe to In His Image. He's putting out some great stuff. Doki Doki Bible Club, the Super Sticker Master. Good to see ya. Thank you so much. I appreciate it. George, yes, it's been a wild night. We had Sal giving part 2 of his lecture series on protein probabilities. So make sure you go check out not only part 2, but check out part 1. We're going to have him back next week. I'm looking forward to it. So George says, Sal killed it today. Lecture and after show. Healthy competition. Amen, brother. Amen. Andrew Graham, good to see you. Good to see you. Yeah, we're going to have a lot of fun tonight. I'm looking forward to this one. And I took a couple sinus pills because I have been sneezing all day. I don't know what it is. I guess we're just going overtime on this whole creation versus evolution thing. But there was no way I was not going to stream tonight because this is exciting and important. So let's get right into it then. Because this video that Dr. Dan, as we can see here on the screen, it was quite hilarious, I'm going to say. And you know what? They say laughter is the best medicine after all. So Dan apparently went into his video blind. It was a blind rebuttal, as in he never watched it prior, which, to be honest with you, was incredibly apparent since he literally, and you guys are going to see this, he literally ignored the content of the video itself that he was supposed to refute. And the point of the video was opposite of what he was focusing on, because he favored repeating his typical talking points that he has been essentially repeating since he got onto YouTube. The video he was responding to, guys, it was a rebuttal to the portion of Dr. Swamidas and Erica's video where they misrepresent. But since they have yet to correct themselves, at this point, it is just, I'm sorry to say it, I mean, they're just lying about Dr. Jeanson by saying he ignored other pedigree-based studies on the why, which I absolutely demolished, and they need to admit it, or else they are admitting that they are purposely being dishonest about Jeanson's work. So Dan, you guys are going to see this. I'm going to play the video. I'll be playing it at 1.5 speed so we can get through it. But Dan quickly realized he could not defend their straw man arguments. And it was so funny because, as you guys are going to see, he literally gave zero defense for Erica and Swamidas and debunked literally nothing. Actually, this comes to mind. Dan's video, and if anybody in the chat can come up with some better memes than me, then go for it. Maybe we'll make this into a competition, because Dan's video, I think, could result in a ton of hilarious memes. His video reminded me of, let's think, him coming home for dinner, okay? And his wife asks him about his day, and he starts ranting about Dr. Jeanson and pedigree versus phylogenetic rates, or she asks him about his day, and he starts talking about his day 3 days ago. Or somebody asks Dan, what's your favorite movie? And he starts listing off all his favorite books. Or, hey, Dan, what's your favorite Marvel superhero? And he starts listing off a bunch of DC comic superheroes. It's like opposite of what his response should be. So, guys, point is, Dr. Dan, at this point, MVP of Team Dodgeball, he's kind of a troll. He's not really to be taken seriously. It's why essentially he's blocked from so many big-time creationist channels. You've probably noticed he really just repeats the same 4 or 5 already debunked talking points and asks the same questions that have been answered a million times. These guys aren't looking for answers. One that comes to mind, look at my Neanderthal debate recently on Modern Day Debate, okay? Both Walker and Dr. Dan in the chat could not follow what I was saying about patriarchal drive, okay? And the fact that the longer you live, the more mutations that'll be passed on, of course. The more that will accumulate in one's lifetime. Somatic cell line mutations, they're not inherited, but more will accumulate in your germ cell lines, and they'll be passed on. So here's the thing, and I was explaining in the debate that if Neanderthals were founded by an early biblical patriarch, okay, as the book of Genesis would suggest, and the evidence suggests, well, they would have started off with what? A massive number of mutations. Plus, as I indicated in the debate, they were earlier. They were an early branch of man. They would have started off different. This is just common sense. They would have started off with different sets of biodiversity. They would have started off with more diversity. I mean, it's not really that hard to comprehend. But remember, when they only have 1 or 3 talking points, they can't really accept an answer, okay? So that's the point. And here's another thing too, because Dan said, yeah, Godzilla Freak, good to see you, brother. Dan says anything, SFT says, you said more things wrong in that sentence than there were words, and it's true. That's what, typically one of their 20-minute videos can result in a series of videos because you have to address every single word. So Godzilla Freak, you've been paying attention. George Bond, Doki Doki, thanks so much for the super stickers. Let me, I'm going to share a screen real quick, because here's the thing, guys. I was, Dr. Rob Carter, he's an expert in mitochondrial DNA, okay? He's trained heavily in genetics. Now, Dan is guilty of this too. They all are. They try and discredit Dr. Carter and say, oh, he's a marine biologist, don't listen to him. And none of them are aware that Dr. Carter has actually published in the mainstream journals on what? Mitochondrial DNA genetic diversity. He's an authority on the topic. And in the comment section of my Neanderthal debate on Modern Day Debate, a few people were making that argument. They were repeating Dan's argument. And I said, do you realize that Dr. Carter has published numerous papers in creationist journals, but they won't accept that. But he's also published in mainstream journals. And they said, no, I didn't know that. And a few of them were surprised and happy to look up Dr. Carter's work. And they found that, the specific paper on mitochondrial DNA. One critic who I'll say deserves a spot on Team Dodgeball, I pointed out the fact that Dr. Carter's published in mainstream journals on mitochondrial DNA. And then he responded, oh, I'd like to see that, condescendingly. And I didn't respond because I was like, do we have to do all the homework for the evolutionists? Just Google. I literally, before we went live, okay, like I was saying in the chat, I said, I'm just looking at the paper. So I saw this atheist comment. I'm like, let's just see how much effort it takes to find Dr. Carter's paper. I literally typed into Google, mitochondrial DNA - Rob Carter. And it was like the third link that popped up. I mean, do we have to do all the homework for these guys? So I'm going to share a screen and just show it, just to put this one to bed. Okay. Quit using fallacious arguments, address the data. Okay, it's a red herring when people like Dr. Dan try and say, oh, you know, don't take that argument seriously because Rob Carter has a PhD in marine biology. It's a red herring. It's a rabbit trail. And I'd like to know how many peer-reviewed papers on mitochondrial DNA Dr. Dan has published. Let's see. I'm going to share a screen, you guys. If you guys are interested, I want you to see the paper I'm referring to. So right here. Dr. Rob Carter, mitochondrial diversity within modern human populations. Okay. Mainstream journals. One day I may actually go through this and just make like a stream on this paper, but highly recommend. He's an authority on the topic, which is why people like Dr. Dan can't even begin to address his arguments. How many times do I show clips from our Rob Carter debate? Because we brought Dr. Carter on about a month ago and had Dr. Carter address every single one of the critics' arguments. There's been no response. We might as well title it the irrefutable interview. This one as well. Dr. Dan and Walker, he repeated Dr. Dan's arguments about unrooted trees versus rooted trees. And I've directed them to this paper multiple times, and I still suspect that it has not been read by the critics. So an overview on the independent histories of the human Y chromosome and the human mitochondrial chromosome. So this is a lengthy paper as to why the mitochondrial DNA phylogenetics points to a young Eve, low variation in mitochondrial DNA and Y chromosome. So one day, I think we're going to do like I'm going to start a series, I think, soon, because we're almost done destroying every single sentence of these guys' arguments, Team Dodgeball. So I'd like to start a series where we pick a paper, okay, and go through it. And this would be one of them too, especially the papers that the critics ignore, okay? So there's that paper. Let me show Doki Doki. Thanks so much for the super sticker. Nebuchadnezzar, good to see you. Good to see you. Nebuchadnezzar, you're pretty famous. Okay, I thought you lived thousands of years ago. Catherine Stormgard says, why is Dr. Carter considered an authority? Well, he has studied extensively. He's got a PhD in marine biology, but he's studied extensively in genetics. He's run many technical simulations with Mendel's accountant. But like as I showed here, I'll share a screen again. He has published in mainstream journals. So right here. Check this out. I think my screen's sharing. Catherine might have just got here. I'm not sure. So mitochondrial diversity within modern human population. So Rob Carter has looked at this extensively. He's published in the mainstream journals. He knows what he's talking about. And his work has not been debunked. So essentially, this is how the critics read these technical papers that are recommended to them, blindfolded. It's got to be the case. Okay, so let's go directly to Dan's video. We're going to play it for a bit, and I'll make my comments, guys. So this should be fun. I haven't rewatched it. I watched it 5 days ago when it first came out, and I wanted to respond that night. I was so eager, you know. I don't require 3-4 months to respond to these videos. Now, we've been so busy with debates, interviews, discussions, editing videos that I just haven't had a chance. And even today, we did a lecture with Brother Sal, and we're going to be busy all week doing stuff. But I thought, you know what? Let's get this out of the way. This is too much fun. And Tuesday, we're going to have George Bond and Steph back on. Steph gave a global flood lecture. maybe a few weeks ago, and we're going to debunk some more portions of Erica's video. But this time, so since we've dealt with all the genetics, now we're going to get into the geology, age of the Earth. They said the hardest part of their video was Joshua Swamidas's interview. So I thought, you know what? Let me start with that. And I think I've got about 9 or 10 videos on that section. So share audio. And let's see. So we got some good stuff for you guys. Let's go right to Dan's video. That's why I've started it at 5:35. Okay. For the main fact that his first 5 minutes was mainly the introductions and not really anything important. So playback speed, let's go 1.5. Now, guys, we figured out how to avoid the echo. Now, me, I am guilty, I admit. I'll play 10 seconds and I'll go on, because like Godzilla Freak pointed out, oftentimes they say more things wrong in one sentence than words that are in that sentence. But what I'm going to do, I'm just going to let it play for a few minutes. I'm going to mute myself so I won't be able to talk, but at least you guys won't have that echo, okay? So let's go back here. Go to my stream. I'm going to mute, so you won't hear me from here on out, guys, just for a few minutes. And
because I've talked about these arguments before. The argument that Jeanson makes when it comes to his pedigree rates, and he does this for mitochondria, and he does this for Y chromosomes. The argument he makes is you can look at parent offspring, and you can look at the number of mutations between them, and you can determine a per generation mutation rate. That's called the pedigree mutation rate. And what Jeanson does, he says, okay, that's the one generation rate, so that's a certain number of mutations. Now let's look and see how much variation there is among all of humanity. How long would it take to accumulate that level of variation given our one generation rate? And then you could work backwards and see how many generations it would take. And by doing the math that way, Jeanson arrives at a comically young age for the mitochondrial and Y chromosome most recent common ancestors. Now, the reason that math is wrong is that you can't just take that one generation rate and project it backwards, because you're catching all sorts of things in that rate. You're catching somatic mutations, mutations like in skin cells, right? I'm going to have mutations that are different from my children because my somatic cells have mutations and their somatic cells are going to have mutations, but those don't get passed on. There's also selection in the germline. So if you have a mutation that causes the gamete to not work, then that will never be propagated to subsequent generations, but you can catch that stuff in pedigree rates. And then over many generations, you've got natural selection, you've got genetic drift, you've got population bottlenecks. All of these things are filters that weed out mutations over time. So what you see is that you start off, if you measure your pedigree rate, it's very high, right? Your parent to offspring rate is very high, but over generations, that rate decreases and it asymptotically approaches what's called the substitution rate, which is the rate at which new mutations are fixed within the population, right? And that takes many, many, many generations to decline till you get to that rate, right? And when you're doing coalescence analysis over many, many generations, like you're doing for the Y chromosome and mitochondria, you need to use that substitute, that longer term rate. You're not using the per generation rate because it goes through many steps of filtering, essentially, where you lose mutations that occurred and then they don't propagate, right? So that's why this rate is wrong. So I'm interested to see if there's anything new here that is different from everything we've heard before. I suspect it will not be. We're going to find out. So I've got this video queued up right to where it gets into the content. And oh, actually, I do need to be listening. It would be good if I could, hear the video. And please let me know. I have it set up that you should all be able to hear the video as well. Just let me know if that's not the case, and I can fix things. So I'm also going to play this at 1.5 speed because it's short, but we don't have time for doing this at slow speed. We're going to do this at 1.5. So let's do it. Let's see if there's anything interesting in here.
all right, let's unmute myself. Okay, so let's comment on that. He brought up a lot there, but here's the thing. Everything he talked about there did not, and I see the chat. So Godzilla Freak says pretty clear so far. So yeah, to avoid the echo issue where the video picks up my microphone, what I'll do is I'll mute myself, play the video, then I'll make my comments, which means we'll play a few minutes each time. So I'm not back and forth with dead noise like that. So, but here's the thing. We have done a ton of videos, okay? Hours and hours and hours on mitochondrial DNA pedigrees, right? Pedigree rate versus phylogenetic rate, predictions. The video that Dan is supposed to be refuting has to do with Swamidas and Erica claiming as one of their main arguments that Jeanson apparently has ignored or missed many Y chromosome pedigrees. So you're going to see that he was expecting to go into a totally different video. Therefore, I am going to address a little bit about what he said there. But since it doesn't really have anything to do with the video he was supposed to be refuting, I would direct you guys to our probably 30 plus hours worth of videos debunking Dan's claims there. Okay, so yes, Dan, yes, evolutionists, we do like the empirical method because we like empirical high confidence science, okay? In pedigrees, okay, which Dan has such a major problem with, all we do, and I've said this many times, all we do is compare rates in the present. We use parents to offspring. Then when we compare mutation rates between parents and their children, okay, what we now know is that the mutation rate is much faster than the phylogenetic rate, which assumes deep time and ape to man evolution. It's rather simple. Evolutionists like Dan, though, they don't want to use the fast mutation rates because it conflicts with their deep time evolutionary story, okay? So here's the problem. Now, what Dan is saying, yes, okay, and CRISPR has stated this actually in Erica's video as well, that by definition, the fast mutation rate that we get from pedigree studies obviously means that the overall mutation rate after many, many, many generations is going to be slower. There's no contesting this. We all agree. The reason why this is because selection is going to weed out a lot of these mutations over time. A lot of these mutations, they're essentially going to be lost. But that is not the point. And Dan seems to miss this. The point is that when we actually consider purifying selection, substitution rates, genetic drift, bottlenecks, all the things that he mentions typically, the amount of mutations we see in either the mitochondrial DNA or the Y chromosome are far too few for deep time evolution. They really, what they do is they invoke fancy stories to account why there are so few mutations, okay? They can't make predictions. Dan attempted to say that he's made a prediction, and it turned out to be a retrodiction that Matt and I addressed in a couple hour-long video. Not to mention, though, that the fact that mitochondrial DNA worldwide and Y chromosome worldwide are what? They're incredibly similar. There's very little variation, meaning we descend from two common ancestors, mitochondrial Eve and Y chromosome Adam. They've termed him Y chromosome Adam according to our model, and based on the fact that there's only about 4,500 years worth of mutation accumulation in the Y chromosome, it would be Y chromosome Noah, okay? And these common ancestors of all people on the planet, they lived just thousands of years ago, not hundreds of thousands of years ago. And this is what Dan ignores, this is also accounting for substitution rates, bottlenecks, drift. Dr. Sanford, Dr. Carter have figured this out, and they talk about over and over again in their papers that they allow for a ton of purifying selection. And I also talk about this in my book where I go over, actually, I think I have it here. Let's see. What I'll do is screen share a portion of my book here. Like I said, this portion is not going to be crazy extensive because I want to get into the meat and potatoes of his video. But since he started it off with that, so I'm screen sharing here, guys. Make sure that you can see it. Okay, so here's my Independent Origins book. Let's see. Okay. A few well-known critics of Dr. Nathaniel Jeanson's book, Replacing Darwin, have continuously invoked substitution rates, as Dan is, as a means to explain away the data suggesting our most recent mitochondrial DNA ancestor was just 6,000 years ago. Actually, Dan pretends like he doesn't remember it, and maybe I'll screen share it again, but TJ Parsons of the Parsons paper, we emailed him, and he challenged all these critics that think he had bad methodology that derived at the 6,000 year date in the mainstream journals. This guy's not a young earth creationist. He challenged them. He said he's addressed all the critics. He's sick of it. Okay. Maybe I will show it again later because it's a very interesting reply. And he pretty much challenged all these critics. If you have a problem, submit your peer reviewed paper, Dan. Okay, and Parsons even admitted in this email that if he were to run the same tests and the same numbers today, he'd come up with the same date. Okay, anyways, that was a rabbit trail. So here, what are substitution rates exactly? Substitution rates are the rates at which genes in the population spread to fixation. These militant critics have pointed out that if you get mutations from pedigrees, right, as Dan is saying here, the pedigree studies are those studies that confirm a young date for Eve, since they involve empirically observed mutation rates. The mutation rates will start out really high, as I said earlier, right? And the reason for this is because you get a lot of mutations. You measure things that will get weeded out over time. Dr. Herman Mays, he's one of these critics. Same with Evo Grad. Dan keeps repeating them. They say if you measure the mutations of parent to offspring, you are not getting any mutations that were lethal and resulted in the death of the zygote. These critics have held very adamantly to the position that what we need to use in order to get accurate mutation rates are substitution rates. They say will be used for phylogenetics. This is what they use for phylogenetics. One of these critics has admitted that when we measure mutation rates in pedigree-based studies, the rate is very high. But many of these, and this is essentially what Dr. Dan was saying, many of these mutations will be weeded out over time, which in turn will even it out in the long scale time of evolution and will eventually equal a substitution rate. They accuse us, the creationists, of overestimating mutation rates by not measuring it accurately with substitutions. At this point, it should now be obvious to anybody that evolutionists have become desperate in their attempt to refute the genetic evidence for a mitochondrial Eve. So here's the thing, as I stated earlier, and we can go on and on about this, of course, but my book here does address all these arguments ad nauseum. We have accounted for, let me see here, I think I got this pulled up in one of my PowerPoints right here. Okay. Y chromosome. Let's go through this real quick, guys. This is Dr. Carter, Dr. Sanford, who have analyzed this. We have used SNP data to analyze the Y chromosomes of several hundred men from multiple modern human populations. The analysis has allowed us to reconstruct the original Y chromosome atom sequence, just as we did with the mitochondrial Eve. Like I said earlier, we have Eve's sequence. We know Eve. And Dr. Carter has published in mainstream journals on mitochondrial DNA. The Y chromosome Adam sequence has in turn allowed us to determine how many mutations separate modern men from Adam. Based on what we now know, how fast the Y chromosome mutates, it's better to say Y chromosome Noah. Today, the Y chromosomes of modern men are only about 300 mutations removed from Y chromosome Adam. So here's the thing. Y chromosomes have very, very little variation. Every single Y chromosome on the planet, male Y chromosome, are nearly identical. There's no highly divergent, highly mutated Y chromosomes. I've been asking for an answer to that question because there are some observations we should see if their answers are correct, but we don't see it. We see that the Y chromosome mutates fast. It does some pretty crazy things, but yet every single male Y chromosome is nearly identical. That means it's young. It's not ancient like the atheists or the evolutionists would have us believe. Here we go. They touch on this here. If the Y chromosome mutates extremely rapidly to explain the 70% difference between chimp and man, how is it possible that all men have nearly identical Y chromosomes and are so very similar to Y chromosome Adam? Even if we assume a normal mutation rate for the Y chromosome, about 1 mutation per chromosome per generation, we would only need 300 generations to get 300 mutations. This is the most straightforward application of the molecular clock concept. The numbers are in perfect agreement with the biblical perspective. However, in 100,000 years, right, the evolutionary perspective, we would expect about 100,000 mutational differences between modern men and Y chromosome Adam. About 333-fold than is actually seen. All those things that Dan wants us to consider, we consider them, and it's not even close to where deep time evolution would need the Y chromosome data to be, okay? They have to invoke all of these unrealistic rates. It's not high confidence science, and they can't make predictions. Okay, so right here, this calculation assumes a typical human mutation rate. If we assume the human Y chromosome, there's an article here they refer to. Let's see, had an enormously higher mutation rate than for other human chromosomes, the problem would be vastly worse. In terms of the Y chromosome, the evolution model completely breaks down at all levels. The point is that the biblical time frame fits perfectly with known human mutation rates in the observed divergence from the Adam sequence. So we've gone over a lot of this. This is good here. The evolutionist problems get massively worse when they invoke an ultra-high mutation rate for the human Y chromosome, as necessitated by the new chimp to human sequence comparisons. Guy, go check out, it must have been a few days ago, where Matt and I, we decimated the arguments against the Y chromosome chimp and human dissimilarity. So check that out. This new data is showing that Y chromosome Adam very consistently fits with the biblical perspective. So we've got, and same thing with mitochondrial DNA. We can allow, guys, for a ton of purifying selection. And here's another thing that comes to mind, okay? It's not on the slide here, but Swamidas does the same thing. They have force-fitted the data, okay? They've retrofitted it. They didn't predict any of this. But they try and say that, you know, based on coalescence theory, we would have expected the one Y chromosomal ancestor, the mitochondrial DNA ancestor, when in fact that is not true, okay? Because coalescence theory, and I bring this up frequently, but we never get an answer to it, because there is no answer. This is a fact. Coalescence theory proposes that given enough generations, what they'll say, you're eventually going to be able to trace back to just two common ancestors, when in fact this theory that they propose has many assumptions, but one of the worst assumptions, okay, has to do with random mating. That's where you have a population where all the people have a strong likelihood of mating in marriage. But we know this is not true historically, okay? Humans have existed in tribes that have been separated. There's no random mating. And here's the problem. Everything we see in genetics did not have to be true for evolution to be true, okay? But it had to be true for biblical creation to be true, because this is exactly what we predicted. God tells us he created two people, Adam and Eve. We would have expected this exact kind of data. So let's finish here with a couple of things he said, Dan said there, about Jeanson. Okay, so I want to point out, I'm going to stop sharing here, then we're going to get right into the bulk of his video, okay? Ramat in the house, good to see ya. Okay, good. So everything sounds good. All righty. Yeah, we like high confidence science. So here's the thing. When it comes to Jeanson now, let's get into his arguments about Jeanson. But what's funny is what I'm addressing here didn't even have anything to do with the video. So let's go through this real quick, leave no stone unturned, and then we're going to see him dodge, duck, dip, dive, and dodge the entire video that he's supposed to rebut. But he wanted to start it off with this, so unfortunately, we're going to have to take the first 30 minutes to address it. If you want an incredibly detailed refutation of what he's saying with Matt and I, where we go through every single pedigree-based study, go into the playlist, Refuting the Critics, and you'll find it all there. Okay, so when it comes to Dr. Jeanson, he looks at the mitochondrial DNA mutation rate accepted by what? That's right, the secular community, which Dan says arrives at the young age for mitochondrial Eve, okay? Then he throws in the Y chromosome. This video is about the Y chromosome. Dan says you cannot take a one-generation mutation rate from pedigree studies and project that backwards because you're catching somatic mutations. So basically, Dan is using the rescue device that he used when he first started that we've addressed over and over again. And that is because somatic mutations, right, the ones that build up in your skin cells, okay, the ones that are not passed on, they build up in our lifetime. And they can be faster in some, slower in others. Okay, and supposedly this negates the accuracy of pedigree-based studies, which is ridiculous. Okay, somatic mutations, we all agree, they're not inherited. De novo mutation rates take this into consideration and remove anything that they consider maybe somatic. Like, this stuff's been figured out. You know, he's essentially just waving the data away, and he's dodging. We've considered these things, the purifying selection. I've gone over the mitochondrial DNA phylogenetic tree. You see a pattern that tells us we all came from one woman. There's not a lot of mutations there, okay? And the molecular clock, you're seeing random mutations in people over generations. And you can see that based on the phylogenetics, they can't assume the human to chimp split. Okay, so I would just say look at somatic mutation rates. Feel free to do so. Look at spontaneous mutation rates, substitution rates, genealogical mutation rates. It doesn't matter. They all agree with the biblical creation timescale, and they all corroborate each other. Nothing helps deep time evolution in regards to these observable studies. We like the empirical method, which he scoffs at. Parsons, okay, I went over the Matt uploaded the Parsons portion that I went through. His email response to us, where he just laughed at these critics. Even Dan in the comment section the other day said, Parsons paper, isn't that 20 years old? And it's just so funny because this is a published paper that is contrary to his worldview and his position, that all he can do is scoff. Like Parsons said, go submit your peer-reviewed publication proving his data wrong, which he's not going to do. He just hangs out in blogs all day. Parsons, Howell, all of these studies that we've looked at, they took somatic mutations into consideration. I can't believe Dan's still using this argument. They excluded them from their pedigree studies. So much for that rescue device, okay? I hope he doesn't use it again. It's getting exhausting. And when Dan talks about bottlenecks, I laugh so hard because clearly Dan isn't aware of the fact that these bottlenecks were not predicted according to the evolutionary model, as I talked about earlier. These were all post hoc ad hoc after the genetic data came out that suggests we came from a very small population based on the low genetic diversity in human beings, okay? This was all retrofitted to explain the data. What about fixed substitutions? These match exactly what we would expect if young earth creation were true and the biblical model of ancestry. We see few fixed total substitutions. Once again, they can't address the data. Dan says, so another thing he said there, Dan says you cannot use pedigree rates over time because when you do, these numbers, they match the biblical creation predictions. But guess what? If they match deep time evolution, you can bet yourself that he would say you can use these. But since the pedigree rates prove that humanity is young, and the average generation times, guys, range from about 247 to 504 generations ago, okay, from all these pedigree-based studies. Notice this, guys. Notice that these numbers do not even remotely help deep time evolution. That's why they have to invoke unrealistic mutation rates, unrealistic substitution rates. Their made-up last bottleneck was 200,000 years ago. Some say there was a bottleneck 70,000 years ago. Some say there was a bottleneck with Homo erectus. Some say there was a bottleneck with early man. They're looking at the genetic data and they're trying to retrofit it. So there's not really any there's not really any agreement. Even a lot of people are moving away from the Toba extinction, saying that never actually happened because of the inconsistencies. So here's the thing. In favor of evolution, let's say the average generation time was 35 years, let's say. That's thousands of generations ago. I mean, it's not going to help their case. They're nowhere near, even if you're generous to them. Here's my conclusion on what he said there. People like Dan hate these mutation rate studies. They hate them so much that they'll do anything they can to slow them down. And people like Dan here reach to studies that try to forcefully obtain slower rates, okay? One, they're looking at only one of the or two of the hypervariable regions in the D loop. They're not looking at the full-length regions. Okay, studies should always look at full length, and Ramad talks about this. We talked about this in great detail, where studies should always look at the full length HVR1 and HVR2 sequences. Another thing, okay, is that they're only looking at small study groups from large populations with little diversity. This is why the Parsons paper is so great and why Parsons is so confident, challenges all the critics. Send in your peer-reviewed study. Or they only investigated, let's say, identical twins, single family lineages, or inbred families. Heteroplasmy was ignored, or they place a generation time at a very high age. They add coalescent simulations to the rates, trying to make phylogeny and pedigree rates converge. They're calibrating the dates. they'll rule out any mutation they assume to be somatic as that slows down the rate. Whatever they do, though, it's nowhere near where they need to be. It's really, really bad for evolution, guys. It's not looking good. I think it's about time that I could go on and on for hours and hours on this one, to be honest with you, but it doesn't even really have anything to do with the video that he's supposed to rebut. So it is what it is. He's just repeating stuff. Matt says, yes, by looking at just one region of the D loop, like HVR2, you do not see heteroplasmy. Yeah, that's right. I mean, Matt wrote a whole book on mutation rates. I mean, this stuff has been dealt with. Brother Matt, you just got here. So when we first started, we went over some papers from Carter, one of his secular papers on mitochondrial DNA. You know, I'm going to play this video real quick as a way to end this, okay? As a way to end this and show you why Dan is just getting really, really desperate. Then we're going to get to him dodging the video, and you guys are going to laugh. So I'm going to mute myself here, guys, so there's no echo. Okay, we're going to let this next clip that I'm going to show you, we're going to let it play through, and then we're going to get right back to Dan.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
They have an evolutionary creation model, which makes no sense. No, in creation, God can front load the genome with as much diversity as he wants to. If all variation is caused by mutation, absolutely, it's impossible. You can't get all the variation we see in modern people if we start from Adam and Eve, if they're 100% homozygous.
Right.
But what if God front loaded Adam and Eve with like 20 million variants? Then we can lose half of them and still have as much as we have today. And 20 million is not a lot. I carry 3 or 4 million. You carry 3 or 4 million of the common ones, and probably 3 or 4 million rare ones. That's fine. So it's actually trivial to start with Adam and Eve and get the people we have today as far as the number of variants. Especially if you had 6,000 years of mutation, you can have a lot of new variants appearing in that 6,000 years.
Right. They're coming from their perspective of the evolution having to work in a short time period.
Yeah.
Well, so what are some of the best evidences for a literal Eve and a literal Adam?
Well, the best evidences are the fact that we found Y chromosome Adam and mitochondrial Eve. Now, the evolutionists put them in a different place where I would put them, but it's quite clear there's only one human Y chromosome. And all men share a very similar Y chromosome. That didn't have to be true, because if we came from a common ancestral population with chimpanzees, that ancestral population would have had a diversity of Y chromosomes. And when the population split into the lineages that led to humans and chimps, it's possible that we could have, like, in this ancestral population, type A, B, C, and D. Well, if humans have type A and B, and chimpanzees had type B and C, that means there would be a human and chimp who both had type B. They're more closely related to each other than a human is to another human on the Y chromosome. But it didn't work out that way. It's, mathematically and theoretically possible, but it's totally not true. There's only one Y chromosome. In fact, just yesterday, or maybe the day before, they redid the Neanderthal Y chromosome, which is really weird to me, because for a long time we only had female ancient DNA. All the Neanderthals, all the Denisovans, as they're sequencing, they're all female. Like, why is no males? We got like 20 females and no males. Something really weird. But they finally published a partial Neanderthal Y chromosome several years ago, and it was way different. I mean, way out in left field. Like, oh wow, that thing's really different. Well, just a couple days ago, they replaced it with a very modern human one.
Wow.
And the Y chromosome common ancestor between Neanderthals and humans, modern humans, is much more recent than it was last week. They totally redated it.
Incredible. Wow. Where can we find that study? I want that one. But so how about molecular clocks then? Do they support a literal Adam and Eve? And is there a constant clock? Is there an average clock that can actually be made?
Yes and no.
Okay.
If you take a constant average for things we can measure in the laboratory today, you can get an approximation of how long ago Y chromosome Adam or mitochondrial Eve lived. It's only a few thousand years. You don't need tens or even hundreds of thousands of years. Now, they don't like doing that, so they're like, the Y chromosome right now, the clock is grounded in the time when Native Americans got to North America.
Okay.
In fact, the Y chromosome guys, they call that a sanity check that appears in several papers. So they're not using genetics, they're using archaeology to give them a clock so they can figure out how far long ago mitochondrial Adam was, Y chromosome Adam was.
Oh, yeah.
Wow, that's interesting. But if you look at the measurable mutation rate from one generation to the next, it's a lot faster than they want it to be. But I don't like the molecular clock idea. I don't think it works. If you look at a Y chromosome family tree and you look at people that are closely related, maybe in the same group, well, some of those people could have twice as many mutations as their cousin. or relative that came from the same founder of that group.
Mm.
So like group R1b, I'm an R1b, 80% of Western Europeans are R1b. If you look at the R1b founder and then you measure the branch lengths of all the individuals that are R1b, there are people twice as many mutations as, wait a minute, that means there's no clock. That means you can't put your finger on the tree and know how long it took for these many mutations to accumulate. And yet, if you just do a rough approximation, everything is young. There's another issue. I wrote an article called Patriarchal Drive. I published it in the Journal of Creation. Did some computer modeling. I said, we kind of know that the older a father is, the more mutations he passes on to his children. And I said, but if Noah was over 500 years old when he had Shem, Ham, and Japheth, and that population was reduced to 6 people, that means those 3 sons, their father was over 500 years old when they were born. That means they each got a huge dose of mutations. And as that post-flood population starts growing, these really old men are going to continue to have children. And the older they get, the more mutations their children are going to have. I call it patriarchal drive. And what it does is it totally messes up the average. It takes, some kids are being born in this population with 800 mutations, and some kids are being born to a young father and they only have 10 mutations. So you can't look at the branch length on the tree and know that the branch length equals time. It does settle down after a while when now, men today aren't having children when we're 500-600 years old. We tend to have children when we're 30 years old. That's a huge difference. So there are problems with molecular clock, archaeologically, genetically, philosophically, mathematically. Yeah, we can still use it if we want to.
Okay.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Now that you mentioned that, because we know humans have incredibly low genetic diversity, like you said, and you're right, they don't like to point out the fact that the allele frequencies and how rare they are. When it comes to them attempting to explain the low genetic diversity, Dr. Carter, it looks like they look to that out of Africa near extinction event. Is that out of Africa near extinction event, is that plausible? And what are some of the major problems associated with that, since that seems to be what they look to explain the diversity?
Well, first of all, it's a post hoc answer. No one predicted the out of Africa. Out of Africa event before they found the data, and they're like, oh, what do we do now? Oh, well, what if our population is reduced to an effective population size of about 10,000 individuals? Now, it could have been reduced to 1,000 or 100 or 10, yeah, but they don't allow that in their models. They only go down to about 10,000-ish. And what if that stayed in that size for a very long time? They never say how long, but a very long time. Well, then, according to mathematics, you're naturally going to have a Y chromosome atom. You're naturally going to have a mitochondrial leave, and most genetic diversity will be lost. The small populations, the inbreeding, you get a lot of loss of diversity. That's why we have, breeds of dogs. It's because they don't let them breed with another breed. There's only, I don't know, how many German Shepherds in the world? How many St. Bernards in the world? Not many. There's a few thousand of each. I don't know. I don't know the numbers, actually. That'd be an interesting question. But that will dictate to you how much diversity you have. The problem with this is this. There are about 10,000 cheetahs in the wild today. And all the population wildlife ecologists are worried that they're going to go extinct because mutations are increasing, birth defects are increasing, litter size is decreasing, reproductive incompatibility, genetic diversity in cheetahs is going down, 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 down. They're too inbred. If they can't survive about 10,000 individuals, how did we? And if that inbreeding event is a near extinction event, we evolved from the primitive Homo erectus to Homo sapiens in a situation that should have driven us to extinction. If you phrase it like that, all of a sudden you realize, like, wait a second, this doesn't make any sense. I mean, we went from half monkey, half man to flying to the moon, right? In a very short amount of time, going through an event that should have bred us into extinction. Yeah. So, it all depends on which syllable you accent in the story.
Well, I agree with you 100% that it's a post hoc, ad hoc rescue device because they never predicted it, like you said, Dr. Carter. And I've heard. Never predicted it. Never predicted any of this data. For anybody who just got here, here's Dr. Carter's paper on mitochondrial DNA. So mitochondrial diversity within modern human populations. 99.9% of the critics, they read these papers blindfolded, or they don't even know about these papers. They just repeat the same argument. Oh, Dr. Carter's a marine biologist. I mean, this guy's an authority on the topic. Here's a paper that, like I said, we're going to go through line by line one day because I reference it a lot. And based on the parroted arguments, I can tell that they haven't been addressed. So here's the Y chromosome phylogeny and the mitochondrial DNA one. The tree that I show, I've got my own specific slide on it, but Dan keeps using the unrooted, right? The unrooted versus rooted tree. And he doesn't want to explain the data. He wants to ignore the data, ignore why there's so few mutations, okay? When in fact, read this paper, read Dr. Carter's work on it, and if you place the root on the evolutionary assumption, you don't necessarily have the same number of mutations along the resulting branches. That's what I always point out. This breaks the assumption of a molecular clock, which is the basis of where they place the root. Okay, and in this paper, because Dan, when I show my unrooted tree, that I show the pattern, you know, explosive growth from a center point, for example. The Y chromosome is the same. The fact that there's so few mutations, okay? The whole point is it being unrooted. In other places, you can see Dr. Carter indicate where the evolutionary root is, and he discusses what happens when you root the tree to the evolutionary assumption, but they're not up to date on the literature. They never read the papers that I cite. I mean, do they want me to just go through this 20-page technical paper during every single debate to prove it to them? But then they'll say, oh, you know, this is a published paper, but this one's in a creationist page. Okay. We got mainstream ones too, guys. We've got it all. We've got it all. So let me see the side chat, and then we're going to continue with Dan's video. We're now getting to the fun part, okay? Okay, so let's see. Thank you, Doki Doki. I appreciate it. Good to see people here. Nebuchadnezzar says, interesting. Yeah, definitely. We our full interview with Dr. Carter is about an hour and a half. And we made sure to ask him all the critics' best arguments, all the objections. So definitely go watch that interview. It's not going to, as this video proved, it's not going to stop them from repeating the same arguments. That's for sure. But we know it's been dealt with. We know it's been dealt with. Okay, so let's go back to Dan's video. I'm going to mute myself again, guys. Okay, and we will allow it to play for a few minutes because I'm going to be muted, and then we'll address it. Okay, here we go. Now pay attention to how Dan, when he starts to see what the video really is about how Erica and Swamidas. Now, if you want to see, so what's nice is Erica did like at the end of her 3.5-hour video, she did like a section where she kind of broke everything down. She reiterated her Swamidas interview that I think goes on for an hour in just a couple minutes. So that's what this video is. We put together a 10-minute video for the critics who don't want to watch our 2-hour videos. But if you want to see every single detail of Swamidas and Erica's claims, I've also got the 2-hour version. So you can do the 2-hour version. And I put in the description box, it's funny when Dan can't even represent a 10-minute video correctly. He's not going to be representing a 2-hour video correctly. And we're doing this for them, giving them short videos because they're always complaining about the 2-3-hour videos. They can't even deal with the short ones. All right, here we go, guys. I'm going to be on mute. We'll let it play for a few minutes. Pay attention. Pay attention to Erica's arguments. See what Dan responds to. He's going to go back to the pedigrees again that I just addressed. That's not the point of this 10-minute video, Dan. Like I'm almost worried about him. I think the beatdowns that we've been giving him in his arguments, I think I don't know. You can see it in his face though, where he's thinking like, ooh, I shouldn't have done this one blind. This isn't going to look good. So, because the video is very specific. We're refuting very specific claims of Erica. And that's the issue. He was incapable of having a rebuttal to it because it was so specific. So it's almost not his fault. He should retry. Okay, here we go. Yeah, Renwell says it's hard to track down arguments in huge videos. Yeah, that's why we've been putting out a lot of 3-minute, 4-minute, 10. So, for example, the TMR4A argument that Swamidas uses, time to most recent 4 alleles. I did a video that's over 2 hours on it, broke it down into detail. But then I released a video that just a 5-minute video on it the other day for people who don't have time for 2-hour videos, or the PRDM9 gene that helps in recombinational hotspots. I've got a couple-hour video on that, but we also released just a 2-minute video on it. So, you know, we're trying to help. We're trying to help everybody out. who might not have. Thank God I found this whole 2x speed, watching these videos at 2x speed. Now I'm going to be able to get through so many more of these videos. Okay, here we go, guys. About to mute myself. Let's listen to good old Dan.
We observed that there are about 50 single nucleotide mutations per
Please let me know if you cannot hear the video. It should be good, but let me know.
generation across 6 billion nucleotides of DNA, which is almost spot on to the evolutionary mutation rate, if a bit too low. To support the average gracious mutation rate, in contrast, we would need to see 5,000 to 10,000 mutations per generation. This is just far more.
So this is by the way, this is Guts Gibbon that is being shown within Standing's video. So this is we're starting with the video that Standing is responding to.
Actually observe. Jeanson makes a case, though, that his far faster mutation rate is indeed correct, and he does so by referencing 2 papers that he does as the only 2 comprehensive studies on the subject of Y chromosome mutation rates. Quote, to date, 2 published studies explicitly attempt to obtain the pedigree-based per-generation mutation rate for the Y chromosome. Both studies have reported the results to be consistent with the evolutionary timescale, unquote. Of course.
So first thing, just responding to Jeanson right there. This is silly because, again, it's a pedigree rate. You can't use pedigree rates over long periods of time. We've been through this. We don't need to rehash it. Pedigree rates are not appropriate for calculating long-term convergence times. It's just using the wrong tool to do the job.
As we discussed earlier, this is simply not the case. The problems with Jeanson's methods of getting a faster mutation rate and thus a shorter timescale are threefold. Firstly, Jeanson's referenced high-quality studies have woefully high error bars. This is due in part to what Dr. Swamidass was discussing earlier with regards to coverage being paramount to a good analysis. Jeanson's papers have pitiful coverage when compared to other studies concerned with pedigrees, meaning Jeanson's own methodology and requirements of no evolutionary assumptions. Sally, for example, in 2016 performed a review that included 11 pedigree papers, all of which were consistent with evolutionary theory and an ancient timescale. Additional reviews have shown that by the time Jeanson released his paper in 2019, there were already countless papers on the Y chromosomal mutation rate both alone and with the rest of the genome. Second, he forgets to subtract out the error when he computes the rates, which is what gives him the high mutation rate, which in and of itself renders his work on the subject entirely erroneous.
The survey said.
I do just want to just pause right there. All those problems with Jeanson's work are correct. Those are valid criticisms of Jeanson's work. He cherry picks his data. He ignores more recent studies. He ignores larger, higher quality data sets because he can't use them the way he wants. In fact, he exploits those high error bars in other contexts as well, like when, for example, he's calculating generation times. He'll use the big generation time error bar for his calculations and then pick the lowest number on there to get an average 15-year generation time, which is nuts. So now I think we're getting into the actual SFT part of this. And by the way, if you didn't hear me say this before, I'm doing this at 1.5 speed because it's too slow if you do it on normal speed. So sorry if that's fast for you. I apologize.
Dr. Nathaniel Jeanson goes through 4 Y chromosomal mutation rate studies, 2 of low quality, 2 of high quality. Guts Gibbon links to the same Scally review that Dr. Joshua Swamidass has mentioned in his blog.
Hang on, let's go back because I want to see if there's overlap between these papers here. It's Carmen. No, okay, just checking.
Scally review that Dr. Joshua Swamidass has mentioned in his blog. It's really laughable, but it's also sad at the same time because when you look up these Scally references, and please do, I pointed this out numerous times to Guts Gibbon, so let me reiterate it again. You won't find Y chromosome pedigree rates. You find autosomal ones. This is because the Y chromosome is very difficult to sequence. Why isn't Guts Gibbon? And why isn't Dr. Joshua Swamidass aware?
So what he's saying is when you sequence the whole genome, what you get out of the Y chromosome is relatively minor compared to that. You don't get good coverage on the Y chromosome, and that's true because the Y chromosome is smaller and it has some unique sequences. That's true, but that doesn't. You can go into each of the studies and just compare the coverage. How many times do you actually sequence each base? So how confident can you be in the sequence there? He's bringing up a quibble, but you could just check the coverage and see which ones have higher coverage, and that's trivially easy to do. And then you pick the ones with the best coverage.
Of this, I speak of Dr. Joshua Swamidass since this is where she got the argument. The thing is, the Y chromosome is full of repetitive and heterochromatic DNA. It also shares similarity with the X chromosome. Check out the paper Posnick, al. 2016 in Nature Genetics. It's 1,000 genomes data. But what they did is they wrote a whole separate paper on the Y chromosome because it's so hard to analyze. What I mean is, there's a reason why so few pedigree-based Y chromosome mutation rate studies exist. Anyone who has studied this issue knows this. And of course, if they were being honest, would acknowledge this. Now, it has been claimed by these critics, for example, Dr. Joshua Swamidass, that you need 1,000-fold in terms of high quality versus low quality sequences.
That's just to be clear for everybody here. When he says 1,000-fold, he means 1,000-fold coverage, meaning when you sequence the genome, how many times do you actually read each nucleotide in your sequence? So in general, you want very high coverage. That's how you get high confidence in your sequence. And what Standing is saying is the studies that Jeanson uses have high Y chromosome coverage. Okay, I don't care. It's still a pedigree rate. You'll notice he's not responding to the actual objection here, which is that you can't use pedigree rates to calculate long-term rates of evolution or convergence states when you're looking at different sequences and wondering how long since they diverge from each other. You simply can't use pedigree rates for that. So you could argue over how to get the best pedigree rates all you want. Pedigree rates, which again are parent-to-child mutation rates, those are inappropriate to determine how long it's been since 2 sequences diverged from each other. It's simply the wrong tool for the job. And nothing you're going to say about how to get the best pedigree rates matters when they're not appropriate for that question. I see a question in the chat from Dapper Dino. What does heterochromatic mean? So that's referring to what's called heterochromatin. So when DNA is stored in the nucleus, if it's fairly diffuse, so it's got nucleosomes, which are proteins with DNA wrapped around them, and then you got a little spacer region and then another nucleosome, and it's kind of strung out. That's called euchromatin, and that's where gene expression is taking place. It's relatively diffuse. Heterochromatin is more densely packed, and it's kind of wound around itself and it's supercoiled. And it's really dense, packed up with proteins, so you don't get any actual gene expression from that region of heterochromatin. So regions that are heterochromatic are indicative of regions that are not like active in terms of gene expression.
It's pretty obvious that Dr. Joshua Swamidas could not come up with even a single 1,000-fold mutation rate study, even if I were to offer him $1 million. Only the mitochondrial DNA has achieved 1,000-fold coverage in the vast majority of studies. The reason being is because it's so small. High quality by many studies is actually 30 times. For example, just have a look at the major commercial sequencing companies. Again, these critics, such as Gibbon and Dr. Joshua Swamidas, appear to be out of their league on this one. It has been claimed by Swamidas, who Gibbon gets arguments from, that Dr. Jeanson picked 2 low-quality studies. Again, he went through 4, too high, too low. And Dr. Jeanson showed that the low quality gave evolutionary mutation rates.
One, he's not defining what evolutionary mutation rates mean in that context. 2, I hope everyone caught the ad homs there, and I don't know if you're familiar with SFT, but he's a fan of saying, no, I don't like ad homs. Well, he just drops a lot of just kind of snide remarks for no reason for somebody who claims to not appreciate ad homs. So I just wanted to point that out for everybody. What you're seeing here is not a response to what Gibbon said or what Dr. Swamida said. What you're seeing is insulting the two of them and saying they don't know what they're talking about without actually addressing the thing that they said. So I just want to point that out.
But it was a high quality that fit the younger creation timeframe and fit the biblical model of ancestry. Now, just when you think it can't get any worse for these critics, it becomes clear that they have not read the work. They have not read the relevant literature, because elaborated on by these critics that supposedly Dr. Nathaniel Jeanson missed a number of pedigree studies. They'll say that he has ignored every study that covered the entire genome, when in fact it has been pointed out that you don't just extract a Y chromosome rate by looking at the whole genome rate. The reason is because of the amount of repetitive and heterochromatic DNA on the Y chromosome. Extracting Y chromosome sequence from a WGS study is labor intensive, of course. Now, you'll notice that when these critics, such as Swamidas, you'll notice that when they're discussing the data and utilizing their arguments, Swamidas, for example, never points to the place in these WGS papers that reveals the Y chromosome rate. It's quite apparent that he's never trying to extract the rate himself. Now, if and when he does, he will very quickly find out that it's much harder than it looks. Once again,
Just again,
All right, let's end it there for now. So isn't it hilarious how, and I'm going to screen share and show you that I've corrected Gibbon on this months ago and still repeating the same argument. So as you can see here, what I'm doing is I'm rebutting the portion, because we've dealt with the pedigree rates where he says you can't get a pedigree rate and get the early biblical date that we look to and that we say, well, I've already addressed that for the first 30 minutes. So he keeps going back to that. It's like his wife saying, honey, how was your day? And he starts talking about Dr. Jeanson's pedigree rates, or he starts talking about his day 3 days ago. It has nothing to do with it. He's not addressing any of the arguments, the counter arguments that I'm using against Erica and Swamidas, because there is no arguments. I am proving that they're flat out wrong, that Jeanson did not ignore other studies. Okay, he didn't address any of the arguments, any of the challenges that I posed to Swamidas. You'll notice that Dr. Dan did not address the Scali paper. He does not admit that Erica and Swamidas were wrong about Dr. Jeanson only using 2 papers. Did you notice that? I showed he uses at least 4, 2 low quality, 2 high quality. Okay, and acknowledges all other studies that are not Y chromosome pedigrees. Okay, he's been proven wrong. And he won't admit that anybody on his team were either misrepresenting, using a straw man arguments, or just being flat out dishonest. Okay, because the ones that supposedly Dr. Jeanson ignored, they're autosomal ones. This is really bad for Team Dodgeball, guys. I mean, he's dodging, he's ducking, he's dipping, he's diving, he's dodging all over the place. He doesn't even address the Posnick paper that I talked about. He ignores the, I guess we didn't specifically talk about it there. I think it's the Balanovsky paper. You'll see, I'm going to screen share you in a second where I've corrected Eric on this months ago, and still repeating the same arguments, not answering my questions. Dan here ignores, but there's no answers to these questions and to my arguments other than them just admitting they were wrong. Or if they continue repeating these arguments, now they're just admitting that they're dishonest or they didn't read Jeanson's. Here, for anybody who just came into the chat, I'm going to show you how Swamidas. Okay, you see this? There's Jeanson's Y chromosome prediction paper. This is Swamidas right here. This is how he read it. Dan hasn't even read it. Erica hasn't read it. They're repeating arguments from Swamidas and Evograd. Here's Dan. We're coming for him. We're coming for him. Watch out. Watch out. The young earth creationists, the arguments, they're coming after you, Dan. These guys got no arguments left over. Okay? This is what we're dealing with. This is their best. This is the best they got. This is why I'm telling you guys. This is why they don't do what Dr. Stefan Frello did. and actually submit these pathetic rebuttals into the AIG Journal where Jeanson can laugh at them and then destroy them. Jeanson has already said he will respond to any of them. Jeanson wishes that people like Evograd and Dan would publish in the AIG Journal with all their critiques and rebuttals. Then he can respond. Here's the thing, in blogs and things like this or comment sections, you can just delete things, change things at a moment's notice, not even admit to your mistakes. No, Jeanson, the AIG Journal, they want accountability. They want the objections submitted where Jeanson can systematically debunk them like he did with Dr. Frello. Just notice Dan here, though, guys. I'm specifically addressing Swami Das and Erica's claims. And then he says that I'm ignoring his argument about pedigrees. Well, I already spent the first 30 minutes debunking that, but that wasn't the point of the video. It's like one ear out the other. This is a comedy show. This is a comedy show. Oh. We've debunked Dan's claims a million times. He still repeats them, as we proved for the first 30 minutes of the video. Keeps going back to that which we've debunked. Dan, this wasn't a rebuttal video to you. I know you think the whole world revolves around you. It was to Erica and Swami Das. I mean, how misinformed can you be, Dan? I'm going to give Dan advice here. Never do another blind video rebuttal, because even your own audience knows that you did not answer anything or even address anything seriously. Man, this is disappointing. This is disappointing. So, you know what? I'm going to screen share, guys. You guys are going to love this. I debunked these arguments months ago. Okay. So screen share. So in a nutshell, Renwill, we addressed why the fast mutation rate in these pedigree-based studies, the observable mutation rate, by definition, after many, many generations, it will be slower because there will be purifying selection and a lot of those mutations will be weeded out over time, okay? That's essentially Dan's argument. He'll say after a long enough time, your pedigree rate equals a substitution rate. But here's the thing. Even if you allow, and we went over the numbers, we went over some papers, some citations, even if you allow for all the things that he wants to, give them generous amounts of purifying selection, give them as much as they want, account for genetic drift, account for substitution rates, account for bottlenecks. Guess what? They're nowhere near their 100 or 200,000 year date, nowhere near it. It's always closer to our date. That's why the mitochondrial DNA worldwide, okay, is nearly identical, extremely low variation in the mitochondrial DNA and the Y chromosome, okay? We know they both mutate fast. So even allowing for all this purifying selection, even allowing for all these factors that we don't ignore, you're still coming up with a young Y chromosome and a young mitochondrial DNA. There's no way around it. Yeah, no problem, Renwell. It's good questions. It's good questions. Okay, so I'm going to share screen. All right, guys. So this was from. Okay, so here's Carter's paper again, for anybody new to the chat, mitochondrial diversity within modern human populations. This is in mainstream journals. Okay, so we're going to go to, this was months ago, guys, right here. I think this is, okay, here we go. Okay, so this was a comment section, okay? Let me know. I'm looking at my phone, guys. Let me know the audio is good. You can see everything. No problem, Renwell. So I'm looking from my phone. Okay, so guys, this is funny, okay? This is where, you know, and I've been giving them the benefit of the doubt, but I just don't know if they're really being honest anymore. They're not answering any of the questions. They're ignoring arguments I've already posed. This is a 60 comment thread on the orphan gene video that we put out months ago debunking Erica, which she didn't even address the orphan gene arguments in her 3.5-hour video. She just dodged it because there's no real empirical evidence for how these novel genes that are highly essential to organisms can come about through any series of mutations. Mutations are going to break things down, not build things up. So anyway, so read through this, guys. This is in the orphan gene video. So a month ago. Okay, so she used the same arguments in the video. So I said, okay, guys, so you can see, Boom. Really pay attention to this. This is important. This is actually going to be the final blow to them, okay? So I point out the mutation rate was because she used the same argument. Jeanson avoided papers. He only used 2. All the same arguments that she used here. I refuted them. Dan was incapable of refuting my refutations. So he went into an entirely different direction and decided to go to his one talking point about pedigree rates, which was nothing to do with the video. Okay. So I say, the mutation rate, Erica, was justified based on 4 papers: Carmen al, Zhu al, 2015 Iceland paper, Moretti paper in 2017. The dozens/hundreds of other pedigree-based papers don't exist. Now, guys, I want you to notice. Okay, I've already addressed this multiple times in multiple videos. I would love to just now debate her on this and ask her and finally get an answer. They're not going to have any answers. Go look at Ramat's and I's video from a couple days ago. Ramat just uploaded it too on the Y chromosome dissimilarity as well as the Y chromosome predictions from the Peaceful Science blog. Ramat and I decimated it. Go check out that video. So I say this is precisely why Gutsick doesn't give references for him. It's been a month and we still haven't gotten references for these so-called hundreds of other pedigree-based papers that don't exist. This is why this is a comedy. I didn't need any time to prep for this. This is child's play. This is a month ago I addressed this. Hundreds of Y chromosome studies probably exist, but they don't test the mutation rate. This is why Erica needs to really stop copying arguments from other people who don't know how to be honest. Dan, Evograd. Erica really should address this major error. She really needs to address it now. That Dan attempted to rebut it blindly and couldn't even correct my arguments and won't even admit that they were wrong. She should either provide the references or explain why the mutation rate studies support young Earth creation. Let's see. Okay, and this one's different about dyads versus triads. That was an oftentimes repeated argument, but I haven't heard them use that one in a while after we corrected them. Here's another one. Erica copying Evograd and Swamidas. Here's the Balanovsky paper. She sent this to me as a reference, supposedly. And once again, it was a fail. And I asked questions here, guys, okay? Doki Doki says, Erica won't fall for our traps of answering questions. Yeah, exactly. Let's see if Dan can answer this one. This Balanovsky paper that you'll see on the Peaceful Science webpage too. So I said, Erica copying Evograd and Swamidas. Go run to them for an answer to this. We haven't gotten an answer, by the way. We have Balanovsky. Answer me this. Why do you think this is relevant to the whole Y chromosome tree? Where does this group of men fit on the whole tree? Let me help you. You will have trouble answering, which we've proven there's been no answer. And Dan didn't provide an answer here in his blind rebuttal because the paper has 2 contradictory trees in it, making it nearly impossible to answer the question. Once someone from your anti-science camp can resolve the tree question, it's likely that we'll find that the branches are unusually short, as the G1 branches are in the 1,000 Genomes Project. I mean, I was detailed here. One of the trees in the Balanovsky paper also shows the branches to be short. The other one doesn't. Hence the contradiction. Short branches imply mutational slowdown, which would make it almost irrelevant to the mutation rate for the overall tree. Guys, look at this. Remember that Scally paper that I corrected her on just now in that video and Dan ignored? Remember? Look at a month ago. Let me highlight that, guys. Let me highlight that for you. Scally. I think Swamidas tried the same trick. It's almost laughable. Guys, look at this. The 11 studies are of whole genome rates, not Y chromosome. Now, let me point out something. Dan wants to pick on the pedigrees, and we can't have reliable pedigrees. Guess what? He's never addressed the successful predictions based on Jeanson's findings from the high-quality sequence papers that we only see 4,500 years worth of mutation accumulation. Guess what? He hasn't attempted to address it. Erica attempted by recycling an argument from the Peaceful Science blog from Evograd and Swamidas. And Ramat and I did a 2-hour video just last week debunking it. They haven't responded to it yet. And then we reiterated it in our video a few days ago. So they're out of luck. We debunked his pedigree argument. He's going to disagree with it. Fine. But here's the next step, Dan. Why are the predictions working so well? Why can't you refute Dr. Jeanson's predictions that are working so well and have led to an active research program? I predict he'll still nitpick. He'll still use the same talking points. He won't read these papers that I've recommended. He'll still keep saying that Dr. Card is a marine biologist and yet won't show us where he, Dr. Dan, Dodgeball Dan, has published in mainstream journals on mitochondrial DNA. I know he hasn't. So here we go. The 11 studies are of whole genome rates. Isn't that funny? But she still used it in her video. Not Y chromosome. Did you know? Look at this. Very simple question. Did you know the 11 studies are of whole genome rates? Or are you just copying again? Scroll down. No answer. So she never gave an answer. Yeah, boom. Guys, go look. If you want to see a funny, I guess, I don't do text debates, but this one was fun. I pinned it 2 months ago, actually. So this one. Evolutionists still don't understand orphan genes. And we're being fair. Guys, we're being fair. Look at this. This is a video that was also ignored in her 3.5 hour long video. Dear evolutionists, science has confirmed Adam and Eve, refuting the... I pinned this. I pinned the same thing here 1 month ago. Same arguments. Look. Right here, down here. Same thing. Scally paper. Month ago. Nope. 3.5 hour video, repeated the same arguments. We responded again for 2 hours. We even put out a 10-minute video to make it easy for them. And then Dan did a blind rebuttal and didn't answer anything. So, oh, it's funny. It's too funny. Okay, let's play some more of his video. Just a second, guys. Let's see. I'm going to do... Here, we'll just do a little bit more of his video. There might be an echo this time, but I want to do... You know, this one's almost too easy. I was hoping for a little bit more of a challenge, but okay, let's keep going a bit, guys. Let me know if I still got the audio on, I believe, but make sure it is, guys. Let me know in the chat. Isn't that funny? Quibbling about specific papers. Oh, the papers that they brought up as a way to rebut Jeanson, as a way to say he ignored papers. I decimated those arguments. And now Dan, damage control Dan, doing what he does best. He just wants to keep going to this pedigree thing, which we've already addressed extensively. I even unnecessarily spent the first 30 minutes of this video addressing it again. But Dan, I get it. You don't agree. You don't agree. It's in one ear and out the other. Okay, fine. Why are these predictions working so well then? Why is the data that we're deriving that you say should not be used, why is it leading to such amazing and successful retrodictions and predictions? And why can't you guys make any predictions? And why, even when we consider purifying selection and all of these factors, why is it still nowhere near your date? You know, why chromosomes? Why are they nearly identical worldwide? Okay, they need to invoke all of these rescue devices, and yet we see no highly divergent, highly mutated Y chromosomes on the planet today. Just, you know, just dodging all day long. Let's keep going. You see the talking? Just repeat, right? Most of those mutations are weeded out. Pedigree this, pedigree this. Pedigree eventually equals phylogenetic rate. We've addressed it all. We get it. We get it, Dan. Come up with something new. What's his PhD in? Dodgeball? You know? If you've ever seen Dodgeball 1, the main stars were Vince Vaughn and Ben Stiller. Dodgeball 2, I want you guys to go congratulate Dan and Swamidas are replacing Vince Vaughn and Ben Stiller. So it's going to be good. It's going to be, they're great at dodging. They're great at dodging. It just shows that people they're not very versatile. His channel is going to die. He deals with like 3 topics over and over again. Junk DNA. I recently responded to that video. Out of Africa. We've dealt with that intensely. Neanderthals and pedigrees. We've literally addressed all his content. But he's not very versatile that he's just going to have to shut down his channel or keep repeating the same stuff. So maybe I sound mean, but at this point, it's just getting frustrating where we're not getting anything of substance, you know, and he's trying to deceive his audience into them. I don't even think his audience fell for what he's doing. They can hear my arguments and then they can see him not even addressing them. I mean, it doesn't take a genius to see it. Oh, and it's so funny. The one that he gave where he says, you know, we can make a prediction, right? We can make a prediction on this, and the substitution rates, you know, they work out in favor for our model. Matt and I did, Matt's in the chat. What was that? A couple-hour video showing why it wasn't a prediction. It was a retrodiction, and it failed miserably. But of course, Dan's going to go back to it. But when you don't get any predictions, you got to pretend. No, Dan, in this video, we focused on Swami Das and Erica's their main arguments in Swami Das's interview for Jeanson's Y chromosome was that he ignored all these other Y chromosome pedigrees. That was their main argument. They accused him of ignoring them. They accused him of only using 2. And we absolutely demolished those claims. They will not admit they were wrong. Dan will not admit they were wrong. And Dan keeps going, hey, Dan, what's your favorite movie? And then Dan starts listing off his favorite books. Not the same topic, Dan, the pseudoscience man. And then he says, oh, SFT gives ad hominems. You know, when it comes to these lousy arguments, all this dodging, dishonesty, yeah. You know, it's not even an ad hominem because he's pushing pseudoscience. He's trying to tell kids non-life evolved into life, and then that life evolved into whales, pine trees, monkeys, banana plants, and humans. there's no evidence for that type of large-scale evolution. He won't take the abiogenesis challenge, and he's misrepresenting Jeanson. I mean, there's no better way to refer to Dan other than what we've been saying. Can you guys hear the audio here? Do I have it shared? Let me go see. Stop sharing. I think I had it. Audio share. Okay, let's go back to it. All right, guys. Here we go. I'm going to mute myself again. We're going to play it some more, guys, okay? Because clearly it doesn't, the sound's not the greatest when I'm not muted. So here we go.
The problems with Jeanson's work, because this is not an expansive takedown of Jeanson's work on this. This is a response to this specific video. But like, I mean, there's cherry picking going on there. I mean, Jeanson just ignores other studies that are more recent and that actually.
Did you guys see that? He literally just repeated their argument that I debunked. That he just repeated that Jeanson ignored other studies. I mean, rip your hair out or what? I mean, it was just debunked in the video, and yet he had no response to the scally. Okay, guys, I'm gonna actually put this to bed right now. Okay, I've pointed this out actually in several videos. Even videos recently, I think in our last video, okay? So, and no response has been given. If Dan would have just did a simple Google search and updated themselves, okay? In 2017, and I actually went over this paper, I screenshot, this just came to mind again now, so I don't have the paper present right now on my computer. I guess I could look it up, but look at Ram Adonai's last video. I actually screen shared the video, went through it. So in 2017, a Nature Reviews genetic paper was published on the Y chromosome. The lead author was Chris Tyler Smith. And guess what? He's a long-standing leading voice in the field. Now, in this paper, okay, they list the pedigree studies in print. And guess what, Dan? Guess what? Repeating the same arguments that I debunked in that video. I mean, didn't even address the Scali paper. He didn't address anything that I pointed out. They don't list in this paper in 2017. They don't list the 18 or so papers that Swamidas and other strawman critics say that Jeanson missed. You know, at first it was laughable, but now it's becoming sad. And these arguments are so clumsy and weak. This all could have just been resolved if they would have just did a simple Google search. And I've already showed them this paper over and over again. So, you know, there's no excuse to saying that Jeanson missed papers, for example, or, okay, here, let me screen share again, guys. Here we go.
Yes, that are incompatible with his desired time frame. It's so yeah. And also just another quick note, just to say that like Swamidas is not familiar with the research on this is just not reasonable.
Just 1 second, guys. I want to make sure that I'm okay. Let's redo that. So I just want to make sure audio is shared. There we go. So now you guys can see why I titled my video the way I did.
If you go over to the Peaceful Science forums, which I strongly recommend, is a really nice place for.
Yeah, he strongly recommends the Peaceful Science forums, where in RAMAT, we did a video a few days ago on the Y chromosome. We went through their arguments on that forum. We went through them, the arguments that they parrot. See how he's recommending to go to that website where they got all these arguments that we debunked? They haven't been responded to. Isn't that funny? A lot of their arguments here are verbatim from that blog with Swamidas. And he says, you know, it's erroneous of me to claim that Swamidas doesn't know what he's talking about. He doesn't. I've already showed you the way that Swamidas reads creationist literature. He reads it blindfolded clearly with these arguments, not addressing the testable predictions. When Erica did attempt to, Ramat and I showed conclusively why they're just attacking Jeanson for not using evolutionary based assumptions, but not addressing why they're working so well. So let's keep going. Let's keep going.
For actual scientific discourse on a lot of these issues. You know, Dr. Swamidas knows his stuff on this.
No, he clearly doesn't, because for one, his TMR4A argument is complete straw man, complete misrepresentation of created heterozygosity. His arguments here against the Y chromosome, saying that Dr. Jeanson missed a bunch of these papers and making these claims about 1,000-fold sequences, I mean, it's not looking good for them. Oh, and with Swamidas, go check out, it's been shared a lot. I think it's almost at 1,000 views. My video I did about a couple weeks ago exposing Swamidas in that Peaceful Science blog. Okay, Swamidas made the most ridiculous argument about genetic diversity and levels of genetic diversity we should find in humans versus animals. And I did a 2-hour response to it. I see it being shared around on Facebook, which is awesome, guys. So make sure you do it. And I've challenged Swamidas to either correct himself on his claims about diversity, or if he's not going to correct himself, refute the content in the video. But these are the types of arguments we're finding. But Dan is talking very, very highly of Swamidas and saying I'm misrepresenting his knowledge. And I'm not saying Swamidas isn't a smart guy. He is a smart guy. But he's misrepresenting and strawmanning people like Jeanson, people like Carter. He's clearly not updating himself on these papers before he critiques them. Because if he did, he wouldn't say that Jeanson only looked at 2 papers and ignored all these other papers and then used the Scali paper, or not be aware of this paper in 2017 that I pointed out, or use the Balanovsky paper. It's just, I don't even know anymore.
Really worked through the creationist literature on this. And not only that, but he goes out of his way more so than almost any.
Yes. Godzilla Freak says, yeah, we should take down some of the larger channels. Yeah, that's why we started, brother. That's why we started that series. Another Evolutionist Bites the Dust. So we've done R and Raw so far, Vice Rhino, Tony Reed, and Professor Dave. So go check out that that playlist, and we're gonna get back to those ones as well. So, you know, Team Dodgeball popped their head back up, and Word on the street is they're tapping out. Erica might be tapping out, especially after she attempted the abiogenesis challenge the other day for a couple hours with us, could not address any of the chicken and egg problems, could not defend it. For $1 million, could not defend it. So I think they might be tapping out. And if this is the best we can hope for in terms of rebuttals to our latest series of videos, then we'll finally be able to just strictly focus on our Another Evolutionist Bites the Dust series. But yeah, go check out the playlist for that series so far. Like I said, we've debunked Tony Reed, Aaron Ra, Professor Dave, Vice Rhino, and we've got another 100 videos saved that we're ready to respond to. So Godzilla, if anybody sees any specific videos from like the big namers that they want us to address, send either Raw Matt or myself the video and we'll get to it. Nebuchadnezzar says echo. Yeah, I didn't mute myself now for this portion because I'm gonna respond to certain things he's saying every 10 seconds or so. So there will be a little bit of a, yeah, actually here's a good thing I should respond to. Renwell says, Erica said she didn't know she was taking the abiogenesis challenge. So here's the thing. The abiogenesis challenge is really just a fancy way of saying, hey, come defend abiogenesis. And here's why I've called it the abiogenesis challenge. It's because, and you're going to see here, Dr. Dan, they scoff at our starting point of created heterozygosity, right? We have a starting point based on our model that God would have front-loaded the original created types, including Adam and Eve, with pre-existing functional DNA diversity. That's where we start from, and it has led to testable predictions. People like Dan, you're going to see him say it in here. He says, oh, there's no evidence for that. There's no real reason to start with the created heterozygosity model. So I've laughed at that. And I've said, for one, it leads to testable predictions. And two, you guys have no other plausible alternative. Therefore, if you're just going to ignore all the evidence for the created heterozygosity model, then give us a plausible scenario for a naturalistic origin of life, abiogenesis. And Erica has no excuse because she debated John Maddox on abiogenesis months ago, 4 months ago. And she's been stating since the 4 months since then, she's been saying that she's been looking it up. She's been studying it. She's looked over John's papers. And that after show was the after show of the debate with John Maddox and Smokey. It was versus, I think, James and Amy. And I was in the chat. I said, anybody who's willing to defend abiogenesis and evolution, come join the after show. And it was a great after show. I mean, Erica was there. James was there. Amy was there. We had this one guy, his name's Randolph, he's like the president of the Canadian Atheist Club or something. And then it was me, Sal, John Maddox, a bunch of us. And it was kind of like a 4-on-4 debate for a couple hours. And, Erica came in there and give me a break. She's been prepping for another chance to demonstrate the validity of abiogenesis for months. And that was just her excuse to try and say, I didn't know I was taking an abiogenesis challenge. It's just a fun way of saying, hey, listen, come defend abiogenesis, because we already know it's indefensible. So quit trying to pretend like there's really any real empirical evidence for a naturalistic origin of life scenario. And Dan, he can come join us anytime. Give us your best evidence for abiogenesis. We'd love it. People like Walker, for example, Walker in the Neanderthal debate. We had a great debate on Neanderthals, and we discussed the data. And we didn't even go the abiogenesis route because he didn't make such a fallacious argument by attacking our starting point. We all have starting points. It depends whether or not those starting points have retrodictions, predictions that flow from them. So that's the whole point. Erica's welcome to come join us any other time. If she feels like she, can do a better job at defending abiogenesis, come join us anytime. Matt, myself, John, Salvador. We've got Dr. Stadler, Dr. Ryan Hayes. We've done a ton of videos on this. So, okay, let's. Nebuchadnezzar said echo. So, I'll let it play for a couple minutes and I'll mute myself, guys. So I'm going to be muted for a second. We'll let Dan talk a bit more, and there won't be an echo anymore. Just a second. Go here. Catherine Stormguard. We appreciate her because, from my understanding, I think she's an evolutionist, but she reminds me of Dessel Drace. We really like Dessel, where they're an objective viewer. They're honest. Seems like they're legit trying to seek the truth. So, yeah, we appreciate you here, Catherine. She asked good questions. Renwell, you too. Good. You've been asking some good questions. You seem genuinely interested. Godzilla Freak says no one can beat the abiogenesis challenge. Exactly. That's the whole point. You know, the honest naturalist will just admit, listen, we understand there's a lot of chicken and egg problems. We don't really have any answers for it yet. We're hoping that there are some answers and solutions in the future, and then kind of move on. Let's discuss the models. You're going to see what I mean. Dan here, he's going to scoff at our starting point, and yet he's going to give no evidence for his starting point. Watch, I'll show you. So I'm going to mute myself. We're going to let him play. We're going to let it play for a few more minutes. I'll be muted though, so I won't be able to pause it every 10 seconds, but we'll address it all, guys. Just 1 second.
Person that I've watched or read or engaged with in this evolution creation discussion more than almost anyone I can think of. Basically, more than anyone I can think of. Dr. Swamidas goes out of his way to give creationists the benefit of the doubt in the context of these discussions. Certainly, he has a lot more patience than I do to assume that people like Jeanson are acting in good faith rather than knowing what they're saying is wrong and saying it anyway. So to say he's unfamiliar with this stuff is just silly. It's not reasonable. It's simply not reasonable.
Yeah. Dr. Jeanson is not ignoring these studies. This is a straw man argument and a misrepresentation. He is going with studies that have actually done a rigorous analysis of the Y chromosome. These critics go through theoretical reasons why people wouldn't look just at the Y chromosome. This is bluffing. Just look at all the published studies that do focus exclusively on the Y chromosome. Just look it up on PubMed. You'll see that these critics, what they do is they shift the focus to the autosomal rates again. They'll claim that it's 50 times less than the Y chromosome rates, as if the two are actually comparable. This is what happens when critics such as Gutsick and Swamidas attempt to speak authoritatively outside of their field.
And finally, his work covers a very pivotal portion of the entire genome, just 0.005% of the mitochondrial DNA or 1.8% being the chromosome.
Listening to these critics, it has also become quite evident that they still do not understand why us creationists look to the uniparentally inherited DNA as the best way to determine ancestry, as the best way to...
I have to pause and laugh there. Why do you only look at the mitochondria and the Y chromosome? Because since they are uniparentally inherited, you have a smaller effective population size. What that means is, in general, you have less variation within a population in those parts of the genome compared to the autosomes, chromosomes 1 through 22, which means you can fudge the data enough to get them to converge at the times you want. But there is no way to get any other part of the genome to converge where you want. That's why they focus on that. And Jeanson actually in his book, Replacing Darwin, basically, he doesn't quite say it, but he strongly implies that you can only do this kind of analysis with mitochondria and Y chromosomes. And that is just so wrong. It's comically wrong. You can do it with the rest of the genome. We do it with the rest of the genome. There are specific techniques to deal with recombination so it doesn't bias your results. And if you screw up and you miss the recombination and you use recombination in calculating your results, you know what that does? It makes your results artificially sooner than they would be. So the number you get in an autosome, if you're doing coalescence analysis, you're doing these same techniques using whatever mutation rate you want to use, the number you get for like the X chromosome, for example, which if I recall, don't quote me on this, I think it's about 500,000 years coalescence time for the X chromosome, most recent common ancestor. So it's outside, it's longer than the Y chromosome and the mitochondrial DNA. When you do that, if you messed up and you didn't successfully account for recombination, that means that your number is artificially recent. So the challenges associated with doing it for other parts of the genome hurt the creationists, because if you screw up and fail to account for those complications, then your number gets worse for them. So you can do this for other parts of the genome. We do it for other parts of the genome. It's just creationists don't like to because you don't get the answer you want. You get more variation, takes longer to converge.
To differentiate between the models, creation versus evolution. Creationists and evolutionists can both agree that the Y chromosome and the mitochondrial DNA, these DNA compartments being the non-recombining DNA, being the uniparentally inherited DNA, we can agree that the DNA differences are the result of mutations over time. When it comes to the nuclear DNA, creationists invoke created nuclear heterozygosity.
So we're going to invoke, so the reason you can't use the rest of the genome is we're going to invoke an unobservable, untestable mechanism that requires divine intervention at some point in the past, possibly after creation, because even if you have lots of created heterozygosity at the start, you just don't have recombination rates that are sufficient, plus the flood bottleneck messes with your amount of variation again. So you're probably going to need either an infusion of genetic diversity, or you're going to need, again, supercharged recombination rates. So this created heterozygosity model is absurd, and I have a video on it, and you could find my video on it. I'm so glad he brought it up here. That's great. I've already talked about this. It's a ridiculous idea that is completely incompatible with reality unless you assume lots of unobservable divine interventions, not just at creation, but since creation. And it doesn't work. There's no way to make the created heterozygosity model work. And Jeanson actually says it's created heterozygosity and natural process, the CHNP model. And it doesn't. Jeanson in his math, he just hand waves away how that recombination math is supposed to work, because he ignores intragenic and specifically intra-exonic recombination. So he's saying you can get lots of variants along a chromosome when the homologous pairs do crossing over. But if you look at the per chromosome recombination rate, it's about 1.2 to 1.4 crossovers per generation. And some of our genes have like 800, 1,000, 1,200 different alleles just for those specific genes. So you would need tons and tons of recombination events within that gene. And repeat that, for every gene on the chromosome, just the rate's too low. It doesn't. This silly created heterozygosity hypothesis makes no sense. But that's par for the course when it comes to creation science.
Which actually makes sense both scientifically and theologically.
Didn't plan that.
Evolutionists assume mutations as the source for all DNA variety, as the source for all genetic diversity.
All right. So because I was muted there, sometimes it's okay. So right off the bat, the uniparentally inherited DNA, like we talk about, the reason why we're focusing so hard on the mitochondrial DNA is because it's inherited. It's uniparentally. It's inherited by female, on the female side from our mothers, and the Y chromosome is on the male side. It's non-recombining DNA for the most part. The recombining DNA, we start with created heterozygosity. And from that starting position, we have very specific predictions that flow from it, okay? We don't just make it up. We have predictions on DNA function, very specific predictions on DNA function that I've covered in great detail in debates, discussions, and other videos. Now, here's the thing, though. As I predicted, no explanation for their starting point, though. You'll see Dan there, as usual, scoff at the created heterozygosity hypothesis. But as always, fail miserably to even attempt what we call the abiogenesis challenge. We were just talking about the 2-hour video where Gutsick Gibbon attempted this. Just attempted. I mean, we call it the challenge. It's just give us a plausible reason to believe that naturalistic origins has any validity to it. So our starting point is front-loaded genetic diversity. And until he can provide a better explanation for the origins of life, okay, he needs to actually address the data, make better predictions, and quit scoffing. Because here's what's funny. Even Swami Das laughs at Dan's straw man argument against created heterozygosity. You want to know why? Because even Swami Das, who's a militant critic against us, he admits that from our starting position, God obviously would have created Adam and Eve and the first life forms with the ability to reproduce and vary. Why would God create Adam and Eve, or the original types of animals, groups of animals, genetically homogeneous? To what? Be fruitful and clone yourselves? That makes no sense. It's a straw man of our model. See, evolutionists oftentimes argue against evolutionary creation, right? Because they're starting with their assumptions in arguing against our position. That's not a good way to dialogue and debate these issues. Like I said, even Swami Das points out the fact that it would be silly to assume that God would have created with the intention to reproduce by cloning. Okay. Now, I've gone over a number of chicken and egg problems for the evolutionists to address, so I'm not going to do it again here for sake of time. But I can recommend, go watch our interviews with Dr. Ryan Hayes, PhD chemist, Dr. Rob Stadler, Dr. Sarfati, PhD chemist as well. We've had a number of interviews, a couple hours long oftentimes, going over the problems with abiogenesis. Dr. Dan has to explain, and I'm going to challenge him then. Explain to us, Dan, in a separate video, how the 4-dimensional genome, okay, the 4-dimensional genome with multiple overlapping codes and chock full of what? Chock full of meta information. We see nested information in the genome, polyfunctional, multifunctional. It's science fiction what's going on a cellular level. Go watch our lecture today with Sal. Explain, Dan, how that came about. There's no human information system that can even begin to compare to the multi-dimensional genome. Okay? DNA can be read both forwards and backwards and have meanings, have function. No book can do that. I mean, we understand so little of the DNA language, and that's why, based on our model of created heterozygosity, we are making predictions on DNA function. While evolutionists expect most of the genome to be the result of evolutionary leftovers, right? Genomic mistakes. So they're expecting a lot of junk, but they're not making any real future testable predictions. We are confident enough to say that based on future genetic knockout tests, extensive knockout tests on mammals, it's never been done on humans. It's unethical, of course, for obvious reasons. But the more tests they do, the more functions will be discovered. Here's another thing I want to point out that sometimes I find evolutionists forget. The genome set of instructions, it's not just a simple linear array of letters, okay? It is dynamic. It's self-regulating and it's multidimensional, as I was talking about. The fact that it's multidimensional and polyfunctional means that these mutations that accumulate from generation to generation, the more functional the genome is, the more deleterious these mutations are. Now they're not so neutral. Now they're effectively neutral. They're nearly neutral. You get a mutation that affects a gene sequence based on the fact that we're looking at a genome that's multidimensional, multifunctional. It is going to negatively affect a whole bunch of things, which means there's shelf lives on genomes, which means the evolutionists are going to have to provide us a type of natural selection that can filter out all these mutations. Here's the problem. Mutations are accumulating faster than natural selection can remove those mutations, right? So now we have a problem of mutation accumulation where selection can only see the worst and can only see the best. But those effectively neutral mutations that escape natural selection, they build up from generation to generation to generation. It is problem after problem, not only for chemical evolution, but also biological evolution. Now, look at the different hypotheses. You got the RNA world hypothesis, that's dead. Protein first is dead. I mean, there are chicken and egg problem after chicken and egg problem. The polymerization problem, the chirality problem, the information problem. Okay. Our DNA is not just chemistry, it's coded information. You get all that chemistry right, now you have the problem of introducing the information. Okay. Information in a book is not just ink on paper. If I were to just spill ink on paper, it's not going to read a coherent message. Okay. It needs to be arranged. The ink molecules need to be arranged in words, sentences, paragraphs. This is where you get the information. Okay. So even if they get the chemistry, the necessary chemistry, how do they get the information, Dan? I'd love to see if there's no plausible scenario in what we know about the chicken and egg problems that abound for abiogenesis. That means naturalistic evolution is dead in the water. And a lot of times they want to say, okay, we'll just give you the first life. Okay, you're just going to give us the first life. You're going to give us an intelligent designer. And then you're going to assume that that intelligent designer just designed the first single cell and that's it? Left the first single cell to evolve into all the life we see today? No, we're proposing, based on our model, that God would have created life forms with pre-existing functional information, and we can make predictions. And guess what? We see epigenetic modification. There are millions and millions of switches in our genome. Just be ready and willing to be turned on and off based on environmental stimuli. Forward-thinking mechanisms in our genome, redundant mechanisms. These require a forward thinker because they are forward-thinking mechanisms. How does evolutionary processes evolve a spare tire? You know, I always use that analogy for understandable purposes. Evolutionists look to a spare tire and a jack in the back of a car. They look at that like it's junk or it's not doing anything or it's redundant. You know, they always say, why would God build so much redundancy or design so much redundancy in our genome? Redundancy is good design. Any coder, any web designer would say that redundancy is good design, and yet that's what we see in the genome. You know, cars are built with spare tires for a reason. You get a flat tire, thank God you got that spare tire. You know, so what What type of evolutionary mechanisms? I asked that to Erica in our third debate. I've asked it over and over again to evolutionists, and their answer that they give, they don't even realize it, but they are now ascribing a mind to evolution. I mean, it's amazing. So this is why I say Team Dodgeball, they have essentially failed, and they've failed miserably. They haven't explained any one of these problems. You know what? I'm going to play a couple clips from this one's shorter, but if you want to see a really good interview on abiogenesis, where we touch on all of these chicken and egg problems, go watch our interview with Ryan Hayes. And these evolutionists, Dan, Gutsick Gibbon, Professor Dave attempted. He didn't know that he was taking the abiogenesis challenge because, like I said, it's more so just like we're calling it the abiogenesis challenge. It's just, listen, defend abiogenesis. Not many are willing. Professor Dave came out with a video attempting to defend abiogenesis. We had Dr. Rob Stadler on here. We destroyed the video. Check that out, guys. You'd love it. We're waiting for Professor Dave to respond, and then we will have Dr. Stadler back on, and we'll respond to that video. So I'm going to share screen. I'm going to mute myself right now, guys, so you're not going to hear me for a bit. Nice to see 20 people in here, even at 4:30. Brother George, $5 super chat. You are on a roll tonight, George. I appreciate it, brother. He says, all knees shall bow, all tongues shall confess. What are evils waiting for? Time is short. Amen. Amen. Salvation's simple. It's easy. It's as easy as walking through a door, taking a drink of water. It's a free gift. Believe on the Lord Jesus Christ and thou shalt be saved. How hard can that be? You know, the Bible says narrow is the way unto life and few there be that find it. The reason why the way is narrow, because guess what? Most people want to trust in themselves. Most people want to trust in their own works. Look at all the religions in the world. I always say there's only two religions in the world, do and done. People always say, why is there so much fighting in religion? There's all these religions. No, you can safely categorize every single religion into either the done category or the do. The do category is all your works, your religions that teach a work salvation, you know. Your Catholic Church would even fall under there. Islam, Mormonism, Jehovah's Witnesses, you know, everybody's looking to themselves. Even essentially your Buddhists and your Hindus are looking to themselves. All those religions can go into the do category. Then you got the done. What's the done category? That is the category of those biblical Christians who have decided to place all their faith and trust on Jesus Christ, who has done the work for us. He did it on the cross. It's finished, he said. You know, it's easy. A little bit of preaching there, though. But, George, that was your fault, brother. So it's easy. It's a free gift. It's a free gift. And a gift can't be taken back. It's free. It's forever. Now, the Christian walk, that takes work. Once you get saved, you're secure. It's a gift. Can't be taken back. If it could be taken back, it wouldn't be a gift. But the walk, the education, the spiritual education, the obedience, dying daily, discipleship, that takes work. So don't confuse your sonship with your discipleship. That's where you might get mixed up into trusting in yourself, and you don't want that. So, all right, let's go to this video. And Lena Powell, I love it. You always got great comments. The chat's awesome tonight. You guys are great. We got a great chat, great audience. Lena says redundancy is needed. This is the backup. What would happen if you have only one alternative to produce the enzymes that are crucial for life? Exactly. You know, they try and use that argument. Erica tried to use that argument and say, well, you know, they have Doki Doki Bible Club. Amen. Doki says, salvation is easy. See here. Yeah, definitely check out the gospel presentation. It's easy. Well, why would God make it hard? He wants people to be saved. He wishes that all would come to repentance. He's not going to make that hard. We can't work to heaven. So, amen. Jungle Jargon says, God is able to keep you from falling. Hey, the Bible says we're made a new creature. We're given the new man. We're given the spirit. We're regenerated. That's why when we fall into sin or we do things that we know we shouldn't do, as Paul says, he wants to do that which is right, but sometimes he finds himself doing that which is wrong. But because we have the new man in the spirit, Paul says, put on the new man. let us not only live in the Spirit, right? When we're saved, we're living in the Spirit, but also walk in the Spirit. Don't walk in the flesh. Don't walk in the new man. So, amen, JJ. Walk in the Spirit, walk in the new man, and you won't fall. You won't fall. So, Lena, great point here. Erica used that argument. I've seen it before. Oh, we've done genetic knockout experiments on mice, they say. And these mice, they were fine. Yeah, because redundancy is good. You knock out one gene, thank God you got a backup gene. But guess what? Knock out gene 1 and 2, and that's going to be detrimental to life. There's a lot of experiments showing that you knock out just the right amount of genes, just the right gene, and it's detrimental to these mice. But redundancy is a good thing. Thank God for redundancy. Required foreknowledge from our forward thinker, our amazing creator. So let me share a screen. Dr. Sarfati, I'm going to play it at 1.7 times speed. Let's see if Dan can address some of these other problems that Sarfati, PhD chemist, goes over. You guys are going to like this. I'm going to mute myself for this one so there's no echo and you guys can enjoy.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Moving on from here, I just recently, we were talking about this before we went live. I just recently read through your chapter in Evolution's Achilles Heels on origin of life and abiogenesis. Now, this chapter, personally, it absolutely blew my mind as to how thoroughly you refuted abiogenesis in a naturalistic origin of life scenario, or I should say scenarios. Can you touch on some of the major problems, for example, the massive chicken and egg problems that abound when it comes to abiogenesis? And of course, Dr. Sarfati, and anything else that comes to your mind on this topic?
Okay, well, see, I'm a chemist. I know something about chemical synthesis. I think most of the biologists don't really know that much about it, to be honest. I think Dawkins is pretty clueless about chemistry. And the thing is, it's not a case of going an argument from ignorance. It's actually an argument from what I do know about chemistry and information theory and the complexity of living creatures. I mean, as you may have seen in the Achilles heel video, for instance, if anyone wanted to make a protein from amino acids, the last thing you'd want in the reaction mixture is water, because water will drive the reaction the other way, because you have amino acids combine to form proteins and they eject a molecule of water. So it's called condensation polymerization. That's a fancy term. So the last thing you want is to have water there because it drives the reverse reaction. So no synthetic chemist is going to have water if he's trying to make a protein. And yet they want to tell me that life began in some sort of primordial ocean. An ocean will only have water, I thought. So that's a big problem. And then you've got things like the amazing information storage system. So even the simplest living cell, it's called the mycoplasma, has about 500 letters of DNA information. But these letters, they code for proteins, but you can't actually decode the information in DNA unless you have a lot of decoding machinery. The problem is that DNA codes for its own decoding machinery. It has the instructions to build its own decoding machinery. So which came first, the machinery? Well, how do you get that without the instructions? Or do the instructions come first, but you can't read those instructions unless you have machinery. So you have to have everything working together right from the start. Otherwise, life could not exist in the first place. It couldn't even get started. And if you can't get started, then Darwinian evolution is dead in the water. It's like runners lining up for a race, they all die on the starting block. That's what we got here. There's no life to start even talking about natural selection mutation because the life is not even there in the first place. And another thing is they realize that the accuracy of a translation of the decoding machines must be incredibly accurate. I mean, Manfred Eigen, who was a Nobel Chemistry Prize winner, he worked out that you have a genome of so many of n number of letters, the decoding must be at least 1 over n in accuracy. So if you have 100,000 letters, the decoding machine must only allow one mistake in every 100,000. Otherwise, you get error catastrophe and it grinds to a halt. So right from the start, the decoding machines must be incredibly accurate. So this goes back to Genesis 1. God said he created everything very good. And now we have modern science showing that things had to start very good. They had to start basically perfect. And what we're seeing is degeneration. See, it could not build up from imperfection to perfection because without an almost perfect decoding system, the thing would come crashing to a halt really quickly. I mean, imperfect decoding machine. Okay, you've got instructions being read to make the next decoding machine, but you've got mistakes because it's not a very good reading machine. So you're going to introduce mistakes into the next generation of machines, which will do a worse job of decoding. which will introduce more mistakes in the next generation, and therefore do an even worse job of decoding. So you're going to get the whole thing crashing to a halt really quickly, unless it starts off basically perfect.
Nice. Well, the naturalistic evolutionists attempt to defend the abiogenesis by purporting that RNA world is a plausible scenario for natural. Is it actually plausible? And if not, could you go over some of the major problems?
Well, RNA world does seem to be the go-to thing, but it's interesting. There's a Dr. Harold Barnhart, who's German, working in New Zealand. He wrote a paper called "Why RNA World Hypothesis is the Worst Theory of Origin of Life, Except for All the Others." So he admits there's huge problems with the RNA world, but everything else is even worse.
Yeah.
Well, see, one thing is, what they tell you is that RNA can sometimes act as an enzyme. So what they're trying to do is combine the coding and the machinery into one molecule. But what they don't tell you is that it can either do one or the other. See, if it's trying to code for instruction, it has to be loose, so it can be the strand will be pulled apart and you can reproduce those instructions. But to be an enzyme, it has to be already combined up with each other. See, so it can't do both. It's either combined or it's not. So the RNA has to make up its mind. Is it going to be an enzyme or is it going to be a coding molecule? It can't be both.
I see.
And then RNA is an incredibly unstable molecule, because I think you might remember 2015 Nobel Prize for Chemistry awarded to Thomas Lindahl and
I wanted to point out that I like how he says it's not an argument from ignorance. It's an argument from biochemistry, from empirical science. There's no plausible or empirical explanation for all of these chicken and egg problems that abound with all of their hypotheses. RNA world, metabolism first. And they hope, dream, and imagine that life will originate in some warm little pond. And they'll say, give it enough time. More time means more degradation. Because real chemistry, as Dr. Sarfati was saying, drives the reactions the opposite direction that abiogenesis would require. So I wanted to point that out. I really like that. So remember that. It's not an argument from ignorance. Okay? It's not God of the gaps. It's an argument from science and biochemistry.
to others, because Thomas Lindahl discovered that DNA breaks down incredibly quickly. It's just very vulnerable to ordinary chemistry. So he realized that living things must have amazing repair machines to undo the chemical damage that you suffer every day. Every day, each one of your cells has about 1 million DNA letters damaged, but you have chemical machines to repair them. Otherwise, you'd be a mutated mess really quickly. You couldn't survive. So this is why the Nobel Prize was awarded in 2015.
Once again, we're looking at more forward-thinking mechanisms in our genome. These DNA repair mechanisms. How many breaks did he say a day? 1 million breaks that are repaired, or else there'd be what? Mutational breakdown, and nearly right away. So, Praise, you there? Okay, you want to stay in the back? No problem. Yeah, so let's keep going. This is too good. I love this. Sarfati kills it here. Doki Doki. Greatest hoax on earth. Yeah. Check out his book. Check out the whole interview with Sarfati. Doki Doki says Dr. Sarfati beats people blindfolded in chess. That's true.
For the discovery of instability of DNA and the needed chemical repair machines. But RNA is about 100 times less stable than DNA.
Wow.
So therefore, the idea of RNA arising in a primordial soup when it's a very unstable molecule, it doesn't make any chemical sense. In fact, even the building blocks are very unstable. I mean, ribose, adenine. I mean, for instance, even Stanley Miller, you may have heard of him, the great grandfather of the origin of life experiments. He did experiments to show that ribose disintegrates in boiling water and even in ordinary water.
So it looks like right from the start, based on what you're saying, Dr. Sarfati, they would have a major issue trying to even get the building blocks for RNA. The building blocks, as you said, are incredibly unstable.
Mm-hmm. Well, for instance, they talk about ribose being formed by formaldehyde in an alkaline solution, but what they have to do is they have to stop the reaction at a certain precise time. Otherwise, there's a reaction called the Cannizzaro reaction. When you have alkali and sugar, it'll actually decompose them. So you have to have someone stopping the reaction before the stuff starts to break down again. And that's, I think, Dr. James Torres talks about this. He's a leading synthetic chemist in the world. And he talks about how any real synthesis, you have to control the time. You must stop it at the right time. You must have the reactions in the right sequence. You must control the pH, which is acidity or alkalinity. You have to control the temperature. All these things have to be controlled. Otherwise, things break down. Primordial soup wouldn't have an organic chemist doing all those controls.
Right. Good point. And aren't a lot of these RNA world experiments highly controlled, highly manipulated, highly contrived?
Well, you see, what they're doing, what they have to do is trying to work out how the first self-reproducing organism arose. So what they're not allowed to use is artificial reproduction scenarios. Like they talk about evolving RNA sequences, but what they're doing is they're artificially reproducing RNA in a way that wouldn't happen in nature to try to explain how the first self-reproducing RNA arose. But they're using reproduction to try to do that, and they haven't even succeeded, by the way. But even their methods don't make sense because they're using what they have to explain to produce. They're using reproductive machinery to explain how reproduction came about in the first place.
How about the carbohydrate first hypothesis?
Carbohydrate first. Well, wait a minute. See, the thing is, carbohydrates, that's just sugars. As I said, sugars are unstable. I mean, that's the thing. They make them in formaldehyde called a Butler-Off or Formose reaction, but you have to stop that at the right place to stop these sugars from decomposing. I mean, that's the thing. Our ribose's instability is very well known. So, I mean, carbohydrate first doesn't really make any sense. And also, carbohydrates, they neither code nor do any work.
Right.
You see, the energy molecules, sometimes structural molecules like cellulose. But you see, in living things, you have the nucleic acid, DNA as the information storage system, and proteins do most of the chemical work. Most of the machinery is proteins, information, DNA. I'm not sure how carbohydrates are going to fit into that.
Right. Good point, Dr. Sarfati. What about somebody who, I guess we got a question here from the audience, I'll throw, somebody who would say, if proteins can last for some time in the water, it's then possible to have them participate in the process. Why is that a problem?
Okay, see, but you can't have it both ways. You can't say, well, we've got billions of years to perform chemical evolution, but with billions of years, you've got time for the proteins to degrade. So the thing is, time is the enemy, not the friend, because the longer you leave it, the more the longer time you have for the reaction mixture to get to equilibrium, which is away from proteins, away from the polymers, and back to the monomers. And the thing is, how do you get the proteins in the first place? Because you can't just, I've got some amino acids from the Miller experiment, which are very dilute and very contaminated. Let's take that to the next stage and see if we can get proteins out of it. They never can do that. Because you know what they do? They say, well, we found a trace of glycine or alanine from the Miller type gas sparking experiment. So let's go to a chemical company and buy pure alanine, which is left-handed. Let's activate and see if we can get some polypeptides happening next. They can't do that. I think this is the sort of step they do. Every time they admit the idea, you've got isolate and purify the material, and then put it into a carefully controlled reaction to do the next step and the next step and the next step. It doesn't work that way. No one's going to spark gases and then try and expect proteins. You'll never find proteins in the Miller type experiment.
I mean, just look at all of these problems. I mean, like he said, it's not God of the gaps. It's not arguments from ignorance. It's just science. It's just facts. It's biochemistry. Wouldn't you just love to see Dan attempt to get through this one with Dr. Sarfati and just try and address these? That's essentially what the abiogenesis challenge is. Answer these chicken and egg problems. Okay, answer the problems associated with the RNA world hypothesis, the metabolism first hypothesis. There's all very fatal blows to every single one of these hypotheses that they purport. Well, no, that's okay. Great answer, Dr. Sarfati. I love the way you put it. That real chemistry goes in the opposite direction than would be required by the origin or any origin of life scenario that says life came from non-living chemicals. Because doesn't water, it tends to drive the reaction in the opposite, or I should say the wrong direction.
If you like, yes. And the other thing is things like left-handed versus right-handed, we covered, like all the proteins in living things are left-handed. My left hand and my right hand, they're mirror images, but you can't map them. I can't fit this right hand to a left-handed glove. It won't work. And see, the proteins and enzymes are all one-handed. Otherwise, the enzyme could not form if you had this mixture of handedness. So the problem is a Miller experiment produced an equal number of right and left, and there's a hard problem in chemistry to try to get one from the other.
So in the Miller-Urey experiment, then, Dr. Sarfati, I still see people use it to this day. Did they find any large biomolecules? Did they find any nucleotides? Anything of significance?
Well, most of what they found was formic acid. Okay, they want to tell you, well, we've got a new way of making formic acid, so we can make new antigen material. That's what it is. So you got a few traces of the of amino acids, and tweaking it, you can get quite a few different amino acids. But the thing is, in the environment they formed in, they couldn't do anything more with them. You couldn't just take that broth that forms and do anything and try and get protein out of that broth. You could never do it because there are too many other things formed that would stop the reaction going any further. Formic acid would contaminate. If you tried to polymerize, say, alanine or glycine from the Miller, the formic acid would get into the chain and stop the chain growing. So you've got these very well-known problems with all the other stuff that's produced in the Miller experiment acting as poison for any further development. They never actually addressed that.
Well, it's a great answer and great points you've made, Dr. Sarfati. Somebody in the chat likes what you said. Time is the enemy of evolution. Well said, Dr. Sarfati. So we constantly hear them saying, well, give us more time. You're not thinking sophisticated, like they'll say, give it 1,000,000 years and eventually we'll get the origin of life. But it's true what you said, right? That more time is no help to them.
Well, yeah, because time means you're going to get to equilibrium. And then you got to look at what the equilibrium is, and it's all the monomers and not the polymers. Equilibrium is the racemic mixture of equal left and right-handed things. See, there's actually a method, it's called amino acid racemization dating method, because they find that over time, after an animal dies, the amino acids and the proteins will go to an equal mixture of left and right handed. That's supposed to be a way of working out how long the thing's been dead. So a long period of time, you're going to get the racemic mixture back, equal left and right-handed mixture back, and you get the proteins broken down. DNA shouldn't last even a few million years, even if it was frozen. And this is actually a lead into another topic we can cover about how we found DNA in dinosaur bones.
Right. Yeah.
Which dated to about 10 times the maximum possible survival time of DNA.
Yeah, that's amazing, Dr. Sarfati. You mentioned it a bit earlier in the fact that, well, for one, I've just got so many points written down from what you're saying. I like the fact that RNA is more unstable than DNA, and DNA is unstable itself. So that seems like a final straw. But the question I want to ask, and I know the audience will enjoy hearing an answer to, the evolutionists, the naturalists, they'll say, well, okay, fine, we don't have an answer for origin of life, chemical evolution, abiogenesis just yet, but that has nothing to do with biological evolution. Can you touch on that? Is it totally different? Or is that a valid argument that they use?
Well, I think it's actually an admission of defeat, really, if they try and say that origin of life isn't part of evolution, when for decades the theories were called chemical evolution. There are departments of universities that are working on this called chemical evolution. That's what it used to be called. Now they say, well, it's not evolution at all, it's abiogenesis, but that's actually a bit of historical revisionism. And the point is, when you look at some of the evolutionary textbooks, I've got examples of magazine like Scientific.
See, Dr. Sarfati makes a great point there. Without any real plausible scientific empirical explanation for the origin of life, then naturalistic evolution is dead in the water. So people like Dan, they never want to address these problems that we constantly challenge them with. But then they want to scoff at our starting position, straw man our position, argue against creation by invoking evolutionary creation, naturalistic creation. You know, that's not our starting point. That's a straw man argument. And if predictions can flow from our starting point, like the created heterozygosity hypothesis, that starting point, and even militant critics of biblical creation, young earth creation like Swamidas, would admit that proposing Adam and Eve would not have been created genetically heterogeneous versus homogeneous, that's a straw man. It makes no sense theologically and scientifically for God to create Adam and Eve genetically homogeneous, which just means no DNA diversity, no DNA differences. They would essentially produce clones. That's our starting point. And guess what? Predictions have flown from it. Predictions on DNA function. And what's funny is those predictions come true more and more every single day with the DNA elements, for example, like the retrotransposons, the ALUs. We know pseudogenes are now showing to be important for healthy life processes in the cell when they've always asserted that these pseudogenes are nothing but genetic mistakes. And we can see that the vast majority of the genome has activity. And Dan will say this activity is just spurious. It's not useful activity. When in fact, that's ridiculous. Why would the cell transcribe all of this useless junk? Natural selection, if deep time evolution was true, would have eliminated all of this junk, all of these pseudogenes, all of these retrotransposons millions of years ago, because it would be wasteful of energy and resources for the cell to transcribe all of this information. Which means this activity suggests it's preliminary evidence for genome-wide functionality. Now we know genetic entropy, genetic degeneration is a thing. So we are expecting some junk, broken genes, of course. So according to our model, we can safely predict 70 to 80% function, right? The gold standard of genetic function tests are what? Genetic knockout experiments. They have not been performed extensively on mammals. So we've got testable predictions in print that are very specific and precise. We challenge the evolutionists to make the same predictions, but they can't because they need to hope, dream, and imagine that the vast majority of the genome is junk. In order for those mutations that accumulate from generation to generation to hit in those junk areas, and then they can be absorbed and become completely neutral, not harmful at all. This is important. And the more functional the genome is, that means there's an even greater waiting time problem when it comes to ape to man evolution. So that's why people like Dan have to say, yeah, there's a lot of activity, but it's spurious. It's not useful. but we've addressed those arguments in detail. Point is, our starting position leads to testable predictions. Now, Renwell says, wouldn't it only eliminate them if they were detrimental? See, here's the thing. If you have a lot of genes that are useless, they're not really doing much, well, the natural selection is not really going to preserve them. Or if they stay within the genome, they would accumulate so many mutations because they're just useless. Natural selection doesn't need to conserve those. But yeah, we see a lot of conserved regions in the genome too. So Praise, don't come in here and ruin my train of thought. You were hiding under the back there, Praise.
How do you like the new background?
Loving it. Oh, did you just do that now?
Yeah.
Okay. All right, let's get more to the we're almost done. Oh, go ahead. Praise is in and out. Okay, just 1 second. I am going to yeah, so especially a lot of these ALU sequences, these retrotransposons, you know, mutations would have mutated them to oblivion, you know, but we don't see that because if they're that functional and essential to the genomes, you know, there's a certain class of retrotransposons. I asked this to RJ Downard in our debate, where in mice, if you knock it out, snip, get rid of it. Well, the mouse will stop developing because its developmental process depends on that specific class of retrotransposons. But the evolutionists say that these all inserted themselves millions of years ago, and then those functions were co-opted. So were these mice just not having children for millions and millions of years until they co-opted the function? The retrotransposon co-opted the function. So, you know, they're involved in gene expression, embryological, and we know so little about the embryological development. You know, what exactly happens on a DNA level? You know, what are each step? So now here's the thing with yeah, good question from Renwell. That's the problem too, is the fact that selection can remove the most detrimental of mutations because selection can see it. And then selection can amplify the best beneficial mutations. We see beneficial mutations. We see sickle cell anemia has a significant advantage. The melanoprotein mutation, for example. Now, still though, beneficial mutations are rare. When they do happen, they're typically reductive, right? Sickle cell anemia has a significant advantage, but it's due to what? Broken cell, broken protein, degraded function. You know, a lot of these beneficial mutations, they're about 1 in a million. Lenski's experiment has told us, but then they're reductive when they do happen. So yeah, it'll amplify the best beneficial mutations, get rid of the work, because right, natural selection is a fine-tuning process, but it's those effectively neutral ones, the ones that selection can't really see, can't really do anything with. They just build up slowly and slowly. Now, no analogy is perfect, but I like pointing out the fact that it's like a single spelling mistake in a book the size of an encyclopedia. You're not going to see every single small spelling mistake, you know, so those will build up over time until that encyclopedia, the message is degraded, for example. So let's go to the rest of this video.
American devoted an issue to evolution and started off with the origin of first life from non-living chemicals by chemical evolution. Okay, so they certainly used to regard this as part of the materialistic worldview. You've got to get a simple self-reproducing cell before you can even start talking about Darwinian evolution. And it's interesting that Anthony Flew, who is the world's leading atheistic philosopher for decades, he was an Oxford guy like Richard Dawkins is, and he said that Dawkins and Darwin overlooked that their theory began with something that had reproductive powers. And this is what a real theory of evolution must account for, to get to this being with reproductive powers. And he says that the findings of 50 plus years of DNA research are a very powerful argument to design. There was this idea, the whole problem with chemical evolution. That's what convinced Anthony Flew to become a theist when he was an atheist for decades before then.
Right. Yeah, that's another good answer. I like what you said there. It's an admission of defeat is exactly what it is, because evolution's dead in the water. You know, naturalistic and evolution and materialism. Well, one last question, Dr. Sarfati, till we move on to a different topic. You mentioned, we were talking about it before we went live, topoisomerase.
Oh, yes.
Could you touch on that maybe for the audience? I think that would be a good topic.
Okay, now the thing with DNA, it's a double helix. You know, it's like a long coiled molecule. I mean, some of you who are older might remember when you had landlines with telephone coils. Okay, I mean, I'm showing my age a bit, I guess. The point is, it's DNA of each of your trillions of cells. You take one of the DNA from one cell. If you lined everything up end to end, it would be about 2 meters long. But it's only a few atoms thick. And you think of coils, they get tangled up very easily. So in fact, living things all have a detangling enzyme and different types of them called topoisomerases. And one class is called gyrase. Okay, what it does, it snips the DNA and does a bit of rearranging and then slices it together again. See, otherwise, DNA would be tangled up and you couldn't actually decode it because it would get tangled up when you tried to decode it, but get tangled up when you tried to reproduce it. See, so without these enzymes working in every living thing we have, DNA would be too tangled up to use. So you think of what topoisomerase means. It has to do three things. It has to cut, it has to move, it has to splice back together. So any one of those processes would not be good enough. You'd have to have every one of them working together. So in fact, one class of antibiotics that you might take if you're really quite sick is called the fluoroquinolone antibiotics. And what that does, it stops the topoisomerase putting the thing back together again. So the bacterial topoisomerase go through the DNA, they chop it up, but don't put it back together. So DNA is chopped to smithereens and the bacterium dies. Right. But then you've got the other chicken and egg type problem is that the instructions that do topoisomerase are coded on the DNA, but you can't read those instructions unless you have topoisomerase in action, making sure the thing doesn't get tangled up in the process of trying to read it. So you've got the thing that the DNA can't work without topoisomerase, but you can't build the topoisomerase without the instructions, which can't be read unless you've got those enzymes already doing their work.
Oh, it's so bad for them. Chicken and egg problem after chicken and egg problem.
Oh, it goes all the way, yeah.
Yeah, it's amazing. I could talk about this all day, and I know the audience could listen to it all day. So I guess before we move on to the evidence for a young Earth, young world, Dr. Sarfati, I am curious, given everything that you've said, are researchers, let's say in this field, are they advancing at all in origins of life research?
I think they're actually finding more and more problems.
Right.
I mean, for instance, we didn't know about the motor ATP synthase, which makes the ATP. It's the world's tiniest motor. I didn't know about this 25 years ago when I first started with the ministry. Okay, so that's something which is, again, a new thing that's been discovered. I mean, we're finding more and more things that make the cell far more complicated than we even knew 20 years ago. So the more discoveries we have, the more problems are being found for evolution. Quite the opposite of what you hear in the propaganda, that science is solving the problem. No, it's actually finding more and more roadblocks to evolution than we had before.
So the more we discover, the worse it gets for them, the more problematic it becomes.
Well, very much, especially when it comes to the origin of first life, yes.
Yeah, that's amazing, Dr. Sarfati. I'll hand it over. Super redeemed energy. God bless, brother. Glad you stuck with us. I know we've been going for almost 2.5 hours, so we'll end it soon. Wanted to leave no stone unturned. So that was a great video. I love Dr. Sarfati, really killed it. And that full interview is a couple hours long. But we took out the abiogenesis section for people to see. So it's just funny, like the more we find out, as he said, the more problems they discover, the more chicken and egg problems. So that's what's funny, you know, how they say, you know, we're just wait till we advance more in science, you know, we'll figure out the problem. No, there's more roadblocks, as he said. So, yeah, it's crazy. Dwayne Burke, good to have you here, brother. Good to have you here. Good to see you as always. God bless. Doki Doki, thank you for the super sticker. Renwill's asking good questions. Yeah, natural selection is best understood as differential reproduction. Who's reproducing the most? Who's having the most babies? That's the problem, though, is there's so many mutations that are essentially bad. They're damaging. They're deleterious. But they don't really have a significant effect on what? Reproductive fitness. So then those people will still have kids. You know, we accumulate 100 new mutations per person per generation. You know, I am essentially more mutant than my parents. I am more mutant than my grandparents, great-grandparents. But I have 2 kids. I could have more kids. You know, I didn't have a mutation that was detrimental enough to the point where I couldn't have kids and I couldn't pass on my genes. Right? It's all about reproduction. Who's passing on their genes the most? If you're not having any kids, you're not passing on your genes. You know, there's a good chance your line, your lineage could be evolutionary dead. So that's the thing. I like, think of it this way. You've got a room full of 100 people, okay? All of them are more or less mutant. More mutant than the generation before it, because we're accumulating more and more mutations every generation. Just get rid of 50% of the worst mutants in that room. Just get rid of them. Okay. Well, you're still left with 50% of the people that are more mutant than the generation before it. So we are going to be consistently more and more mutant. Selection is a fine-tuning process. It can keep the species as strong as it can be, but there's been papers published in mainstream journals, numerical simulations using Mendel's accountant with a variety of simulations. The only rebuttals you'll find are in blogs from like Dr. Dan or YouTube videos, but no real peer-reviewed rebuttals or papers or anything like that invalidating it. And there's been a number of parameters set, and it always comes out, even with like super beneficial mutations. It doesn't matter, because even with a super beneficial mutation, okay, you're only getting an increase in fitness in a very narrow sense, while the rest of the genome is degenerating in so many ways, because these mutations are still pouring in regardless. So these numerical simulations, regardless of how generous you are with evolutionary mechanisms and natural selection, consistently, these numerical simulations show that humans will degenerate to eventually the point of extinction. Now, as biblical creationists and Christians, we believe our Lord and Savior Jesus Christ will come before then, but we can see evidence for the fall in our genetics, genome degeneration. Take this point of most mutation accumulation back to a point of least mutation accumulation, of least genetic entropy. That would be a point of creation, a point before the fall, a point of perfection, a point of Adam and Eve, according to our model. So Dan did not mention gene conversion. You got recombination gene conversion, okay? Can both act to shuffle up these design variants. There's not a problem. You add in gene conversion to the problems that Dr. Dan's talking about, and there's plenty of time to sort out these design variants. Plus, obviously, God would have placed these design variants in functional linkage groups. This is basic. It's a straw man understanding of our model to say anything differently. Here we go. PRDM9 and recombination. Whenever like people like Dan use this, whenever they use this recombination, okay, Swamidas says this, okay, assume created heterozygosity, but how do we get the necessary rates of recombination? Well, I like to just quote them directly from the secular papers, their own literature. The title of the paper is The Landscape of Recombination in African Americans. Let's just read right from their own literature. We have shown that PRDM9 alleles that bind a novel 17BP motif and occur at greatly increased frequency in people of Western African ancestry have led to a shift in the recombination landscape compared with people of non-African ancestry. The larger number of hotspots available so there's a larger number of hotspots, recombination hotspots in West Africans. Because the evolutionists, they'll say, well, there's evidence for more genome scrambling, evidence for more recombination in Africans. And then they'll conclude that must mean they're older. They're older than non-Africans. They're older than we would say Adam and Eve were. But now they're finding that you've got this PRDM9 that assists in recombination, and there's more of those gene sites in Africans. There's a larger number of hotspots. That means Africans have more evidence for genome shuffling and recombination. They've got more diversity because they've experienced more rounds of recombination, not because they are older. The more PRDM9 sites you have, the more recombinational hotspots you have, the better your population will be able to retain genetic diversity. Doesn't mean they're older. So at creation, okay, when we consider genome degradation, when we consider the created heterozygosity hypothesis, logically, at creation, Adam and Eve would have had more of these PRDM9 gene sites, would have had more recombinational hotspots, more effective gene conversion recombination. Effective recombination, they're finding, depends on the more heterozygosity you have. So the point of most heterozygosity would have been what? Creation. Recombination would have been more effective. Gene conversion would have been more effective. This is all easily explainable, but then they want to scoff at it. But then yet they don't want to answer any of their own chicken and egg problems. So if I see Dan use the same talking points and doesn't address the chicken and egg problems associated with his model, his naturalistic starting point, it's proof that he's not even worth dealing with anymore because they're not answering any of the questions. So right here, the larger number of hotspots available to West Africans implies that at the population level, crossovers are more evenly distributed than in Europeans. And thus, the shorter extent of West African linkage disequilibrium is not due to differences in demographic history alone. And I've got a ton of stuff on that right here. PRDM9 influences the possibility of genetic exchange by determining the locations of meiotic recombination hotspots in most mammals. Tons of stuff. PRDM9 controls activation of mammalian recombination hotspots. No problem with allele frequencies. It's so funny because a lot of Dan's arguments have been answered directly in papers that I've cited. Carter, Sanford, they address these problems. I recommend Adam and Eve, Design Diversity and Allele Frequencies. The allele frequencies we see today, it's been, the numbers have been done. They're perfectly in line. They're even better, much better aligned with the biblical creation model. Here's the predictions that have been ignored. Testing the predictions of the young Earth Y chromosome molecular clock. Dr. Jeanson published 2019, December 4th. Population growth curves confirm the recent origin of human Y chromosome differences. So, I mean, paper after paper. You know Eve's sequence right here, the Eve mitochondrial consensus sequence. We know it. Very little variation. You can trace it back to one female ancestor just thousands of years ago. Look at this pattern. This is mitochondrial DNA. Human mitochondrial DNA. Explosive growth from a center point. There's not a lot of mutations here. Evolutionists want to say this is hundreds of thousands of years. Where do you get that? We can account. We can account for this. We can invoke large numbers of purifying selection. We can, all the factors he talks about, we could consider them all, and they're never going to get the data. They're never going to get the data to fit their deep time evolutionary story. Actually, maybe we'll end it with, I should see how the chat's doing, actually. I can't see the chat when I'm screen shared, so. Doki Doki Bible Club, $5 super sticker. Thank you so much. I appreciate it. Godzilla Freak says, we don't stand a chance against the post hoc fossil layer predictions. Yeah. Fossil records, low quality, low confidence science, guys. I mean, it's indirect evidence. The fossil record's highly fragmentary. Okay, they're constantly finding these mixed bone beds. They're constantly, we had Dr. Rob Stadler on last week, and he was going over how they found, I believe it was Tyrannosaurus, and it was different fossils of Tyrannosaurus in the same species, but in different stages of its life. Okay. And, you know, adolescent, adult, and because of the variation in the same species, they accidentally categorize them as different species when it's the same species. So, I mean, and we could go over, I mean, numerous reasons why the fossil. Here's the thing, and I point this out all the time. There's a lot of times there's more variation within the same species than between species. Okay, and the biological definition of species is, can they interbreed? Bones found in the dirt. We have no genetics. Genetics is the best and most direct way to answer this question of ancestry. Without the genetics, you know, without the ability to know if this bone and this bone, if they interbred, then it's tough to tell what's the difference between convergent evolution, where you've got kind of the same traits evolving independently, where in the fossil record, you can accidentally declare recent relationship, when in fact, that's not the case. So, I mean, there's a number of things. We've had video. I have a whole series that I did, 2 or 3 videos, 2 videos, part 1 and 2. I titled it Low Confidence Science, and then a 3rd video addressing objections to it. So, Lena says, Aaron Run. Yeah. I feel like Aaron Ra would be another Todd. We don't want to fall for that trap. Maybe we should play. Maybe we'll end it with a couple funny videos. Oh, here we go. George says, hey, praise, video on oil exploration on Tuesday. Okay, so now that we've really hammered the genetics portions of Erica's latest videos, now we're going to get to what I would say are kind of the easier portions, because they said, like I said, the Swami Das interview is supposed to be the most difficult section to refute. So we started with all of those parts. Now we're going to get into her sections on like geology, age of the earth. So we got Steph coming back on, and George Tuesday. We're going to debunk Erica's arguments on oil exploration and how uniformitarianism is needed, deep time evolution is needed for oil, things like that. So that's going to be a lot of fun. We've got some debates set as well. Doki Doki says, Standing, take your children to X-Men school. Yeah. Yeah. If only, I'm sure evolutionists hope, dream, and imagine that mutations could do what they apparently do in the Marvel universe with some of those extraordinary powers that your X-Men have. Wouldn't that be nice? So let's, yeah. So the recombination, PRDM9. What else did Dan say there? And his objections to created heterozygosity. We've addressed it all, you know, and that was, it was just a sad rebuttal. And I think I've made that evident. I addressed those arguments. Let's end it with a couple videos. Let's end it with a couple videos, couple laughs. Here, I addressed it in my recent debate with Just a Walking Fish, who was, I would say, trained by Team Dodgeball themselves, because especially his arguments with Neanderthal phylogenetics are Dan's arguments that they think are irrefutable. So I'm going to go, here we go, TMR4A. I addressed it directly in that debate. I am going to, so there's no echo, guys. I'm going to mute myself, and then we're going to listen to that too. Just leave no stone unturned on this. And we already know there's going to be no response. So let's have some fun. This has been too fun. With these evolutionists, we got job security for life. Okay, just 1 second.
Please hit that subscribe button, because we are just getting started.
For $5, question for this isn't addressed to anyone, so either of you can comment on this if you like. I have a feeling this is for Standing, just knowing the history of it.
Probably.
Swamidas actually concedes the most recent for allele argument actually takes millions of designed alleles into account. These aren't made by mutation. Explain, please, dude.
Okay, so I'm guessing that's for me.
That's what
yeah, no problem. So this one's a pretty technical one. Let me explain it to the audience. So I like to reiterate the argument as best I can. So this TMR4A, all it stands for is time to most recent for what? For alleles, okay? So this analysis, what it does is it traces the origin of our autosomal DNA differences back in time. And also, though, this is important, okay, because it comes down to recombination. This analysis actually incorporates both recombination and mutation. So the researcher can then follow lineages to the point where this TMR4A point is where 4 lineages are present in the human population. But this is why I pointed out that this model only pretends to assume the created heterozygosity model, because our model treats millions of DNA variants as the result of de novo creation. That means, given the position definition of allele, you're looking at millions and millions of alleles in the created heterozygosity model. So if I were to just accept the terms used in this TMR4A analysis, let me just accept it, okay? Here's the thing. We would be looking at millions of lineages, as I pointed out, in Adam, and millions in Eve. That's why the next question comes down to, okay, how are you going to break up all of these alleles? That's the next question to be answered, and that's where I would point to gene conversion. Okay, remember, more heterozygosity means more effective gene conversion and recombination. And we know the PRDM9 gene, okay, helps with recombination, for example, as I showed those papers. So I hope that was sufficient enough, but good question.
Can I just say something super fast?
Yeah, go ahead.
I still don't understand why the created heterozygosity, you have to put the allele definition like at the base, because the easiest way to get from one nucleotide to another nucleotide is just a mutation. Recombination rates don't happen nearly fast enough for recombination to explain just all of the different lineages on Earth, right? You have like 1.4 recombination events per chromosome per generation. You're not going to get, tons and tons of haplogroups from that. It just doesn't work like that. Also, I'm pretty sure Argweaver, the program he used, does account for like differing recombination rates. You have to like standardize it by using something back through time. I forget how it's specifically done, but yeah, those are just two things.
Yeah, those are some good points. Here's the thing, though, and I read an excerpt from a paper earlier where it shows that just because there's more diversity in a population, it doesn't mean that means that population is necessarily older. Like Africans have more working PRDM9 gene sites, and yet what do they have? Evidence for more recombination. Versus Europeans. Europeans are highly inbred. What happens with inbreeding? You get more levels of heterozygosity or more levels of homozygosity, which would reduce allelic variability. Now, here's the thing: the PRDM9 gene is well known to regulate, control, and influence recombination through hotspots. So, like I said, the created heterozygosity model would assume what? More heterozygosity, which means more functioning PRDM9 genes and more effective gene conversion. Gene conversion can actually split apart these design variants properly and more effectively. I want to point out one last thing. This is why I say that argument only pretends to assume created heterozygosity, because Dr. Carter and Dr. Sanford make it clear in their papers that these design variants would have been created within helpful and beneficial linkage groups. Like, how silly is it to assume that God creates Adam and Eve, he front loads all of these DNA differences, but then, oops, God accidentally placed them in sections of our DNA that can't recombine or can't work with gene conversion. I mean, that doesn't make any sense. So yeah, more PRDM9 gene sites in the past, less mutation accumulation, more levels of heterozygosity means more rounds of recombination, more effective rounds of gene conversion. So I think it's a plausible answer. I don't expect the evolutionists to tap out on it, but I'm happy with it. I think it fits well.
Well, there you go. A short 5-minute. What's nice too is I like debunking their best arguments right in the debates with them. Right now they're there. Now they can dodge, duck, dip, dive, and dodge all over the place, like Dan does in his responses. So those were his objections to created heterozygosity, and as you can tell, there's no problem at all. Okay, what we find is amazingly consistent with our model, and it can lead to predictions. So Doki Doki Bible, you guys are too fun. I mean, we've been going almost 3 hours, still 20 of you in here, but for me it's going on 5:30. I should have had another coffee. So thanks for the super sticker, Doki Doki. This has been a lot of fun, guys. Erica, monkey see, monkey do. Lena, you give Lena, Doki Doki, you guys all give the best comments. Praise with a big smile. Praise is always happy. He's a happy guy. He's a coffee drinker like me. George Bond says, awesome. So Dr. John McKay, geologist, we're going to be hopefully having him on soon. Dr. Jason Lyle. Lots of good interviews, discussions, and debates for you guys soon. I've confirmed a couple. But yeah, no, I think we've addressed everything. That was a fun one to rebut. So what I want to do is I'm going to have a SFT Classics night. So we got about 1,000 videos actually now. 1,000 videos. I can't believe it. Matt deserves a raise, all the editing he does. He's edited 95% of those videos. So if Matt's still in the chat, thanks, brother. You've been a blessing. So we're going to have SFT Classic nights. And Nebuchadnezzar says, not a fan of coffee. Yeah, that's a good thing. It's not good for you. It's a diuretic. It dehydrates you. So it's my one bad habit, I think. Yeah, Dr. Lyle. He said sometime after Thanksgiving. So lots of good stuff to look forward to. Hopefully get Dr. Sarfati back on again. I'll be doing a couple more debates too. Dr. Garrett. He's been patient. He wants that rematch. He wants that round 2 to try and answer some of the questions that he didn't quite have an answer to the first debate. So I think I've been so busy, but I think I'll finally schedule that one in for you. Who wants me to debate Dr. Garrett again? Say aye, and I'll do it. Godzilla says, Doki and Bond fun team at Steph T. It's true. It's true. Without Bond and Doki, we wouldn't be putting out all of these videos. So yeah, I got a whole collection of videos from the big shots that we're going to debunk, and we're going to have some SFT Classic nights. We got 1,000 videos, and a lot of people now, they haven't seen our videos. We made a lot of short, funny videos, especially with Ken Hovind. We've got some good videos of him. with like Professor Dave debunking the evolutionist. So we're going to have SFT classic nights. I'm going to gather maybe 10 or 20 of the funniest old ones that I don't think a lot of you guys have seen. We're going to go live, probably have Raw Matt here, and we're going to go through them, have some good laughs. So Praise says no. We've got a whole playlist of Ron Garrett using his arguments from majority and arguments from consensus. Renwell W. He's maybe triggered over the Ken Hovind. Yeah, I could interact with you guys all night. I should probably get some sleep, 5:30 a.m. So that was a lot of fun. If anybody thinks there's no arguments, some arguments I missed there, let me know. But that really was one of the worst rebuttal videos I've seen. To think it was from Dan, Dodgeball Dan. So Lena says Dr. Garrett is fun. Yeah, we like him. I like him. Him and I talk through email. I'd love for him to come to Christ, you know. Got a whole playlist, Dr. Garrett. Anybody that is new to this channel, they love debates, discussions, go to, even if they don't agree with my position, go to the playlist section. Got a ton of good debates for you guys to keep you guys busy for hours. The last 6 months have been a blast. We put on so many good debates. So definitely check those out for people who are new to the channel. Godzilla Freak says consensus and extrapolation. Renwell says a debate with Garrett again would be cool. Yeah, we like Garrett. We like Garrett. He's there's no reason why we can't disagree but have respectful, cordial discussion. And you know, Dr. Garrett's been a blessing because he's got a PhD. He's been around for a while doing this. He's written on it. You know, he's written on genetic entropy, for example, and he's always willing to discuss these things. He's always willing to debate. I think I've had him on 7 or 8 times, debated Ken Hovind, Bill Morgan, Paul Price from CMI. I've debated him. He's debated Nephilim Free, Salvador Cordova. I mean, he's given us some good debates. Yeah, Kent's been on here for debates about 15 times now. So he's been a bless. Even when I was just a small little channel with 100 subscribers, Kent was willing to come on. So his debate with Derek Barnes was really good. Doki says dogs produce dogs. It's true. That's a fact right there. Nebuchadnezzar says green tea is a great alternative. Yes. Well, my wife bought me some good healthy organic tea called Breathe Easy. Helps with allergies. So I'm working towards better and healthier alternatives. Hans Gruber says, Dr. Ken Hovind is great to listen to. He helps me often. Amen. Yeah, years ago, it was his creation seminar that I watched that opened my eyes to a lot of this. So he's been a blessing, and he's been a blessing to this channel. Like I said, he's always been willing to come on. Even 2 years ago when our channel was small, he's been on for many debates. He's always willing to debate. Okay, so yeah, let's call it a night here. It's been a lot. I can't believe it's been we've done 5 hours of streaming today then between Sal and then Sal had an after show. So no, 8 hours maybe. It's been a busy night, guys. Tuesday, be here. Like I said, we're gonna address Erica's oil exploration arguments and lots more to look forward to. So Nebuchadnezzar says, if you need help with allergies, buy some desiccated. Is that how I say it? Desiccated kidney. It'll help with your histamine. Awesome. I'll look into that. I like natural substitutes. And definitely, yeah, sometimes my allergies act up. So, all right, guys, you guys have been awesome. It's been fun. I like interacting with the chat. You guys have asked a lot of good questions today. And, thank you, Renwell, for asking some good questions. Like I said, we like skeptics and critics here in the chat. We like good questions, good objections. And he says maybe we will help him think a little more positive in terms of creationism. So that's our goal. That's our goal, to show you guys the science and hopefully do our best to address your objections. So anyways, last visual of the day. Dr. Dan's been debunked again. Dr. Debunked Dan. So Everybody, God bless. Stay tuned for some more fun this week. If you didn't watch Salvador's lecture earlier on protein probabilities, please check it out. And SFT is out.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe